Good morning, everybody. Well, good morning here in Brazil, right? We don't know if there are persons that are from outside. I noticed that there is a person from India, so it's not good morning there. So hello, everybody. It's a pleasure to have Dr. Carlos Rinaldi here. He would be giving his last lecture now. Please, Carlos.
Thank you, Andres. So good morning, everybody, and welcome again. Thank you for attending and the opportunity to continue sharing some of our work. So today I'm going to continue this tutorial or series of sessions on magnetic particle imaging. And I'm gonna, for those of you who may not have attended yesterday, I'm only going to show a few slides on what is MPI. It's only going to be 2 slides. But basically, yesterday I provided some background on why iron oxide nanoparticles are interesting in biomedical applications. I provided some background on the history and development of MPI and some of the applications that have been published. Then we talked about the physics of MPI signal generation and how that relates to models of particle dynamic magnetization and particle properties. I presented, I also talked about the effect of relaxation, finite relaxation on MPI performance, and presented some of our work to obtain particles with low magnetic anisotropy and very few defects that are really suitable for MPI. And I think that was it yesterday. So today I'm going to pick up with one more topic related to particle physics and MPI performance, and then I'm going to talk about a couple of applications that we've been working on in my lab and in collaboration with others. So again, MPI, magnetic particle imaging, is a new imaging modality that relies on the nonlinear dynamic magnetization of magnetic nanoparticles. And the advantage here is that the tracer is a biocompatible and biodegradable, typically superparamagnetic iron oxide nanoparticle. The imaging modality is non-invasive, it's tomographic, and it's quantitative. And it has high sensitivity, negligible tissue background, negligible tissue attenuation, long tracer shelf life. And even though it was first reported in 2005 as a concept, there's been rapid development of hardware, and there are 2 commercially available preclinical scanners. And there's also several groups that are working feverishly on developing clinical scale scanners. And so it's an exciting field. It's a relatively new field and an exciting field, especially for those who've worked with magnetic nanoparticles before. This is the distribution of scanners currently around the world. There are 14 commercial preclinical scanners installed around the world, and 5 more expected to be installed this year. I actually think the one at Johns Hopkins was being installed last month, so it might be already up and running, which is exciting. And this does not account for several prototype scanners, for example, at Berkeley, several prototype scanners in Germany and Europe, and also there's scanners in Japan and Turkey. So it's a rapidly growing community, and it's an exciting time to be working in this field. So I'm going to pick up where I left off yesterday. And yesterday I talked about the classical theory, let's just call it that, or the original theory of MPI and signal generation, which is for non-interacting superparamagnetic particles. And so today I'm going to start off by talking about how we can leverage strong interactions between particles to obtain unprecedented MPI sensitivity and resolution. And this is the result of a really fun collaboration with Steve Connolly's group at Berkeley. And there was work done by a phenomenal student in Steve's lab, Ziwei Tai, and 2 great students in my group, Rohan and Shizhuan Shao, who did a lot of this computational and theory work that I'm going to present. So to remind you, when we're talking about MPI performance of magnetic particles, we distinguish between the 2 approaches to generating an image. The hardware is the same, but the way the image is constructed is a bit different in these 2 approaches, the harmonic space and X-space MPI. In harmonic space MPI, when we talk about particle performance, we typically look at the harmonic spectrum characterizing the response of the particles to an oscillating magnetic field of a frequency and amplitude that would be used in the MPI machine. And we're interested in signal attenuation from the third harmonic, and slower attenuation means a better tracer. In X-base MPI, we typically look at a point spread function. And for the point spread function, the peak intensity corresponds to signal intensity in the machine. And so the higher the better, the more sensitive. And then the full width at half maximum corresponds to resolution in NPI, and the smaller the better. And last time I presented some theory that can be used to predict in X-base MPI how the sensitivity and resolution are going to depend on particle size and other properties. Now, as I pointed out yesterday, those models are only for particles where there's no significant relaxation, where there's no interactions, and where the particles have no polydispersity. And yesterday I talked about I talked briefly about the effect of polydispersity, and I talked significantly about the effect of relaxation. Today I'm going to talk about potential effects of interactions. And there's been past work, there's been past experimental work that suggests, for example, that one of the reasons ferrocarbotran or Resovist may be a good MPI tracer, at least better than you would expect, may be because of interactions between the small magnetic particles that make up Resovist that are embedded in this dextran matrix, and they may be interacting. I also alluded to that being potentially important in other commercial tracers. There's been prior work on trying to model or understand the effect of magnetic dipolar interactions on MPI performance. And this is going to focus primarily on computational work, since this initial part of this section has to do with our modeling work. And so, there's been papers that look at randomly distributed particles that are immobilized and that have interactions. And what they looked at in that paper is the effect of interactions on harmonic MPI performance. There's been also work with two particle models where you have particles that are immobilized. And again, these authors looked at harmonic MPI performance. And finally, there's been prior studies as well with randomly distributed particles that can rotate and interact, and that has looked at X-base and harmonic MPI performance. And this is probably not comprehensive, but this is representative of the state of the field when we started working on what I'm going to show you now. And so for randomly, an important thing is that for randomly distributed nanoparticles, it seems based on these papers that strong magnetic dipolar interactions lead to worse MPI performance, which is the opposite of what we want to achieve. And so that might be important. I want to point out that any such predictions have to be taken with a grain of salt. Models are only as good as the assumptions that went into the model. And also the results of the simulations are only representative, perhaps, of the specific situation that was analyzed. And there's a lot of variables that could matter here. And so the fact that there's been this past theoretical work should not be taken as discouragement by experimentalists who may want to explore unique combinations or arrangements of particles that may be interacting. Now, the motivation for our work, and as often happens in science, this went backwards. The experimental results that I'm going to show you at the end were actually observed first. And then we started doing the theoretical modeling, and the experimental work took a long time to finish. And so the modeling was published first, and the experiments are now under review. But basically, in collaborative work with Steve Connolly's group at Berkeley, we discovered that chain formation with strongly interacting nanoparticles, which we're calling superferromagnetic arrangements, lead to improved X-base MPI performance. And so our objective here was to computational study this effect. And we're going to focus on X-base MPI from now on. And so as a reminder, I showed this yesterday. When you have magnetic particles in time-varying fields, they can respond to a magnetic field by either a Brownian relaxation mechanism or a Néel relaxation mechanism, where Brownian relaxation means physical rotation, and Néel relaxation means internal dipole rotation. And within Néel relaxation, you can have uniaxial anisotropy and cubic anisotropy. We can further model these things using the Landau-Lifshitz-Gilbert equation for the internal dipole rotation. and basically a stochastic angular momentum equation for particle rotation. And in both of these models, one can account for the effect of dipole-dipole interactions. And so in the Landau-Lifshitz-Gilbert equation, the dipole-dipole interaction enters as part of the effective field. In the stochastic angular momentum equation, there is a torque associated with magnetic dipole-dipole interactions. And so in this way, we can account for interactions in both types of situation. Now, in the simulations that I'm going to show you, instead of having randomly distributed particles, because our experimental observations suggested that particles were forming chains, we're going to arrange the particles in chains. And the key variables here that can be changed during the simulations are going to be the size of the particles and the distance between particle surfaces. And we're going to calculate average magnetization in the z direction for these particles, which can be used to obtain signal intensity and resolution by analyzing the time-varying magnetization. And we're going to do this in two types of arrangements. One is going to be an alternating magnetic field, and another is going to be a superimposed alternating magnetic field and ramp field. And this is more akin to the type of field you generate in a relaxometer. And so let me show you the results. And so first, the results for alternating magnetic fields. And so here, we're looking at particle size of about 20 nanometers with a surface-to-first surface distance of about 16 nanometers. The chains are going to consist of 10 nanoparticles, and the amplitude of the magnetic field is 20 millitesla, and frequency is 25 kilohertz. And so under these conditions, if you have particles interacting, or responding by the Niel relaxation mechanism, what we find in blue are the results without interactions, and in red are the results with interactions. And so this is the point spread function. You will notice that the peak location has been shifted. So it's not at 0, it's significantly shifted. You will also notice that the peak is not symmetric. But I think the more important observation is that when you turn on interactions in the chain, the height of the peak goes up. And the fact that the peak is shifted can lead to problems in image generation, but it turns out that the Connolly group was able to figure out how to obtain an image out of this, as I'll show you in a few minutes. So this is for uniaxial anisotropy. For cubic anisotropy, the result is even more striking. And so here you can see that with no interactions, you have a very broad point spread function. And with interactions, you see a much sharper point spread function, and also a significant 6-fold increase in signal intensity. And so this is really exciting. The idea that you can have a particle where, depending on whether you have interactions or not, you get such dramatic improvement in signal intensity and resolution is very exciting. Interestingly, the same is predicted for Brownian relaxation. So this is without interactions in blue, with interactions in red, and you see the same effect. There is a change in the peak location, but that can be accounted for during imaging. But more importantly, there is an increase in signal intensity and also a sharpening of the signal. And so this is quite exciting because, again, it illustrates how interactions can be leveraged to obtain better MPI performance. Now I'm going to show you results for superimposed alternating and ramping magnetic fields, which are more representative of the situation in MPI. Now, I want to point out that the Landau-Lifshitz-Gilbert simulations are very time-consuming. And so to generate a scan representative of an actual MPI scan, it would take many years to obtain all the data. So in the previous couple of slides, I showed you the results for both Niel and Brownian particles. But now I'm only going to show you results for Brownian particles, since we don't have the computational capability to model this situation with Niel particles. And so now it's the same thing. Particles are about 20 nanometers with distance of 16 nanometers, 10 particle chains. And the field is, and the bias field is being ramped from minus 40 millitesla to 40 millitesla. And so in blue here is the result without interactions. And in red is the case with interactions. And you can see again a very dramatic increase in signal intensity and a very dramatic improvement in narrowing of the point spread function, which would correspond to an improvement in particle resolution. Here, I'm showing you results for the effect of particle surface to first surface distance on the X-space MPI performance. And this goes to show that by tuning the interactions between particles, which can be controlled through particle size and can be controlled through particle-particle distance, you can obtain different behavior. Also importantly, you can see here, let me focus on this figure first. This is peak signal intensity. And this is the excitation field. And let's look at, for example, the case where the particles are a separation distance of 16 nanometers, which is in red. And what you see is that at a low field, you have a very poor signal intensity. And then if you increase the field from 5 millitesla to about 15 millitesla, you get a fairly dramatic, almost tenfold improvement in signal intensity. And so in principle, these are particles that you could apply one excitation field amplitude and get one behavior, and another excitation field amplitude and get another behavior. And that can be used for color MPI to distinguish, for example, particles that are in these chains and interacting with each other from particles that are not. And the threshold field, as we call it, for that transition from poor behavior to good behavior is dependent on the distance between particles according to the modeling. Full width at half maxima that are predicted also depend on the excitation field. And importantly, the model predicts that we can achieve sub-millimeter resolution, which would be very exciting because until now, the best reported MPI tracer has a resolution, if I remember correctly, of about 0.8 millimeters. And so our postulated mechanism for this is that we're getting this improvement in resolution and sensitivity because of these strongly interacting tracers, because they form, as individual entities, they're super paramagnetic. When you apply a field, they form these chains. Now, when you reverse the direction of the field, the fact that the chains initially have all the dipoles in one direction means that you have to overcome a barrier in order to flip the field. And yesterday we were talking about how maybe with chains you could have some uniaxial type anisotropy, and that's a good analogy here, I think. And we think that that's a reason for why we would get these improvements. So this is the experimental data, and I'm only showing you a snapshot of it. The entire paper is under review, and there's more data in the paper. But this is with 26 nanometer iron oxide particles, and they're suspended in toluene, and we put them in these Lorenzo resolution phantoms. And so here's ferrocarbotran, which is commonly used, and you can distinguish this set of samples, but not these. These are these super ferromagnetic chains, and you can very clearly distinguish all the resolution phantoms, and there's some slight line scans. And so analysis of the point spread function suggests that, and this is in the paper and not in the slides, that we can obtain particles with full width at half maxima as small as 1.51 millitesla. And so if you were to apply a 5 Tesla per meter gradient field, those particles should be able to have a resolution between 150 to 200 microns. And so that's very exciting because that's not something that's been achieved before in MPI. Also, it has important implications to development of clinical scanners, which I'm not going to get into now. Okay, so moving on, the next application I want to talk about is that of using MPI for image-guided and spatially selective nanoparticle thermal therapy. And again, this was in collaboration with Steve Connolly. This is before we got our scanner. And again, it was Z-Way Tai did the experiments, a lot of the hardware development. On Connolly's side, and on our end, Rohan Davalikar worked on theory, and Andreina Shiu prepared particles for these experiments. And so I think Ivkov will talk more about magnetic fluid hyperthermia later today. But basically, what this is a type of thermal therapy for cancer, where magnetic particles are targeted to cancer tumors or delivered directly to cancer tumors and used to dissipate the energy of an oscillating magnetic field, inducing localized hyperthermia. And an advantage here is, again, the particles are, in the absence of a field, not toxic. So there's no toxicity without the magnetic field, but there's significant toxicity when you apply the magnetic field. And it has to be a certain field amplitude and frequency in order to obtain significant heating. This is something that can be used to treat tumors deep in the body. And there's been demonstrated synergy with chemo and radiotherapy, and it also can induce an anti-tumor immune response. As I mentioned yesterday, this therapy has been translated to treat brain cancer in Europe using clinical scale magnetic field applicators, which is often a question people ask if we can do this in a human. And there's really, the hardware is not the limiting situation. It's really figuring out how to do it effectively. And there's several opportunities for further development in this field, such as achieving sufficient nanoparticle accumulation after systemic delivery, controlling temperature rise and achieving reproducible therapeutic doses, overcoming heat transfer limitations in small tumors, understanding mechanisms of anti-tumor immune response, and finally, avoiding damage to healthy organs that accumulate magnetic particles. And so in the majority of the literature on magnetic hyperthermia, what people do is they directly inject the particles into the tumor. And in fact, that's what they do in Europe when they use this as a treatment for brain cancer. And the assumption is that if I inject the particles into the tumor, the particles are going to stay there and I'm going to be able to heat them, and I've delivered sufficient dose. And when we first got the MPI, we were doing a hyperthermia study, and my students got curious and they started to go in and take some images. And this is one of our earliest observations when we installed our momentum. And that is that we would take these mice and they would be bearing orthotopic mammary fat pad tumors. We would inject particles in the tumor. This is a fiducial, so it's a known mass that's put in the sample bed. But then we would also see a signal in the liver, even though we were taking that image half an hour after particle administration. And in fact, we did this for several animals, and we found that in about a third of the mice, the particles rapidly redistributed from the tumor to the liver. And one could see three types of response. One would be all the particles stay in the tumor. Another would be all the particles move to the liver. And yet another would be some of the particles stay in the tumor and some of the particles go to the liver. And the reason this is important is because if we can't deliver and retain a reproducible dose of particles in the tumor, it's going to be difficult to achieve reproducible thermal doses during treatments. Also, if a lot of particles accumulate in the liver, then that can lead to significant liver toxicity if we apply a systemic or whole body magnetic field because the particles in the liver are being heated. You can use other imaging modalities to try to track particles. The iron oxide particles have some CT contrast, and this is a complicated slide, but basically here there's the line, the contour corresponds to the surface of the animal from CT, and the color, the haze is the magnetic particle signal, and we can see a signal in the tumor and a signal in the liver. But in the CT, we see no signal in the liver, so CT is not sensitive enough. But MPI clearly shows that a significant amount of particles have accumulated in the liver. Okay, so what I'm going to show you now, what we did with Berkeley, is we came up, we demonstrate, we didn't come up with it. There's been previous papers for about, I think, 10 years suggesting these ideas, but we were the first to actually combine it with MPI. So we use this concept of spatially selective heating using a selection magnetic field gradient. And this is going to be very similar to what I told you yesterday about the physics of MPI signal generation. But now what we're going to do is we're going to use a uniform alternating field of a frequency such that it promotes nanoparticle heating. And if you apply it everywhere, all the and there's, only apply this alternating field, all the particles heat. And so systemically administered spions would, that accumulate in off-target organs would lead to significant tissue damage in those organs. And this has been shown actually in experiments with mice, and that was from Rob Ivkov group, where they showed that under certain conditions, you could have elevated liver enzymes and liver and spleen necrosis and even animal death. And so the way we spatially select the region of heating is by applying that selection magnetic field gradient. And again, it has a small region in the middle where the magnitude of the selection field gradient is weak. And so the particles can respond to the alternating field. And everywhere else, the particles cannot respond significantly. And so only the particles in the field-free region are able to dissipate heat. And this is illustrated here where we have a 384 well plate with a collection of particles in the middle wells. If we apply an alternating field throughout, we get uniform heating, as evidenced from a thermal camera. But if we put two opposing magnets such that they generate a small field-free region in the middle, we get, and this is the same set of wells, we get heating only in the central wells and no heating in the periphery. And so this illustrates how we could use these selection magnetic field gradients to control the region where particles are heating. So the idea would be that spions accumulate in the liver and target tumors, and we can use MPI to quantify particle distribution and plan treatment, and then follow that up with spatial control of spion heating to achieve selective heating of particles in the tumor while sparing the liver. Here we show this in vitro. And so here we have, again, cells and particles in a distribution of wells in a 384 well plate. And if we apply the magnetic field only, what we find is that throughout the plate, we get fairly consistent heating and also fairly consistent cell kill. Here, the numbers and the color bar correspond to the percentage of cells that have been killed. And you can see that in the vast majority of the wells, we've killed over 90% of the cells. And the reason we don't kill as many cells in the periphery is because of heat transfer losses to the surrounding wells that are not being heated. Now, if we do this in the selection magnetic field gradient, we get a situation like this, where the central wells are heated to the hyperthermia range. The wells in the periphery are not heated to the hyperthermia range. And we see that only the central wells are being heated, the cells are being killed. And the size of this region is actually in the order of a centimeter or so. And so what this means is that in principle, we could control heating at a spatial length scale that is typical of many tumors that would be treated. Okay, and so what we did with Ziwei and Steve Connolly is demonstrate this. And so Ziwei built the hardware using old scanners that were available at Berkeley. And he demonstrated that we can, for example, in a mouse, in this case, the mouse had two tumors. The two tumors were injected with particles, and only the bottom tumor was going to be treated. Here's an imaging scan. During the imaging scans, the alternating magnetic field amplitude and frequency is low, and so it's not sufficient to cause any particle heating. This is the temperature profile as a function of time. In the bottom tumor that's being treated in red, the top tumor in black, no heating at all, and the rectal temperature to monitor body temperature. And what we see is that we were able to achieve a significant therapeutic dose of 79 cumulative equivalent minutes at 43 degrees C. But in the tumor that's not been treated, we basically don't deliver a significant dose. And so this then results in reduced tumor bioluminescence because we're killing the tumor cells in the treated tumor. And so it illustrates how MPI and these selective gradients can be leveraged to enable image-guided and spatially selective nanoparticle heating in vivo. Another topic, which I'm not going to talk about today because I'm actually, there's a pre-recorded talk that's going to be featured tomorrow in the Brazilian Breast Cancer Symposium. But another project that we've been working on a lot is on using MPI to track adoptive T-cell transfer immunotherapies. And so this is excellent work by my student Anjali, and it's a great collaboration with Dwayne Mitchell and a scientist in his lab, Lan Huang Min. But again, if you want to see it, you can watch my recorded talk tomorrow. And so for the last topic today, I'm going to talk about, I'm going to shift gears from cancer and talk about how we can use magnetic particle heating and also MPI. I'm going to illustrate how MPI can be leveraged here for whole organ cryopreservation and rewarming using magnetic cryopreservation agent solutions. And so there is a dire need to improve organ preservation technologies. Our ability to transplant organs has really resulted in prolonged and better quality of life for many patients, but there's severe limitations. Some of the more commonly transplanted organs are kidney, liver, heart, and lungs. And unfortunately, many of these organs never reach the recipient because they exceed a preservation time window after removal. And so, for example, if you remove the heart from a patient, you have about 4 to 6 hours from removal or from death of the patient in order to be able to transplant it successfully to another recipient. And so that is a very limited time window. It makes it difficult to do effective matching between donor and recipient. It also limits the real geographical range over which you can deliver an organ. And so new technologies are needed to extend organ preservation times in order to improve utilization and matching and to reduce the costs associated with organ transplantation. I like science fiction. I love superhero movies, for example, and science fiction movies. And so there's this idea of cryopreservation, which I've seen many times, and I'm sure you all have seen as well. And so here's some illustrations from some movies from the last 20 or so years, where people have been frozen and cryopreserved, and many years later they're brought back to life. And so this seems like science fiction, and I mean that is science fiction. But there is some science fact. In fact, human cells, embryos, and some tissues have been cryopreserved for decades. There's been healthy human and animal offspring that have been conceived using cryopreserved ovarian tissue. And there's also been demonstration of successful transplantation of whole and partial rat limbs and kidneys, rabbit kidneys. And so this can work, but there are significant challenges. Some of these challenges include toxicity of the agents that are used to cryopreserved. But I think some of the more important challenges, especially for organs, seem to be that these cryopreservation agents require very fast cooling to vitrify and even faster rewarming in order to avoid devitrification. Also, although we can cool and vitrify the organs effectively, current warming modalities are limited because typically what is done is that the organ or the sample is placed in a heated bath, and the heating comes from the outside. That leads to significant temperature gradients, which can lead to thermomechanical stresses and damage to large organs. And so enter nanowarming. And so nanowarming is a concept for fast volumetric heating of vitrified cryopreserved tissues using magnetic particles and alternating magnetic fields in order to avoid devitrification and thermal stress. And this was introduced by John Bischoff at the University of Minnesota. They've published really excellent work on this. And the concept is basically you would take a tissue that you're going to cryopreserve, you embed it in this cryopreservation agent solution that contains magnetic particles. You can store it and then remove it, and then in the presence of a magnetic field, you would get rapid rewarming, and you would obtain your tissue without much damage. Now, for this to be effective in whole organs, we need to achieve uniform perfusion of the nanoparticles throughout the organ. And I would argue that nanoparticle stability in these cryopreservation agent solutions has been a problem in the past, and this is some examples from published work. This shows a very spotty distribution of particles in an organ, and here again we see the same using imaging modalities. Now, this is going to happen. It's a new technology, and obviously formulating particles that are suitable for this is going to be a challenge. Nanoparticle stability can have several roles in nanowarming, and so one, for example, is that the nanoparticles must be stable against aggregation in the cryopreservation agent solution for them to remain really small and penetrate deep into the organ, into the microcapillaries. The nanoparticles must also be stable after nanowarming to allow them to perfuse out of the organ. And so these are important requirements. We approach this by designing nanoparticles that have a superparamagnetic iron oxide core, which is biocompatible and is going to lead to efficient release of heat in alternating fields, but it can also be used in magnetic resonance imaging and, importantly for us, in magnetic particle imaging. We also provide a coating, and that coating should provide colloidal stability in complex cryopreservation agent solutions. That colloidal stability should be active in an extreme temperature range as low as negative 196 degrees C. It should remain colloidally stable during rapid heating using the magnetic field. It should be stable in blood, and it should avoid nonspecific cell uptake because after you rewarm the tissue, you want to remove the particles as much as possible. And the particles, of course, should be small enough to perfuse into capillaries, but large enough to prevent extravasation in the tissue. So our approach is to obtain these particles by covalently bonding a dense PEG brush. And I'm not going to show you how we did it, but basically it's a cryoprecipitation method, and we use PEG-silane ligands that are covalently bonded through a siloxane shell on the particle surface, and we can do this at gram scale. And here I'm showing you dynamic light scattering results for particles suspended in the cryopreservation agent VS55. And these particles have no change in hydrodynamic diameter over 28 days in VS55. If you vitrify them and then rewarm them in a magnetic field, there's no change in size. And after vitrification and rewarming, you leave them in the VS55, and even 14 days later, there's no change in size. So these particles are really ultra stable under these conditions. They're also able of ultra fast heating, and so this is now we take about 20 milliliters of this cryopreservation agent solution, either with or without magnetic particles, vitrify it to liquid nitrogen temperature, and either rewarm it at a bath or in an alternating magnetic field while we measure the temperature using a fiber optic probe. And here, the green data is the cryopreservation agent with the nanoparticles being heated in the magnetic field. And you can see that over a span of less than 100 seconds, we go from liquid nitrogen temperature to room temperature. And so that is a heating rate of about 320 degrees C per minute. In contrast, when you have the CPA without any particles, even with or without the magnetic field, the heating is very slow. And the particles alone without the magnetic field, the heating is also slow. That heating rate can be modulated using the strength of the magnetic field that's applied. And also it depends on the concentration of particles. And so this is a fairly easy to control system. Here now I'm going to show you some cytotoxicity experiments using primary cardiomyocytes isolated from mice hearts. And we're going to look at it using analysis of the cells. And so in terms of particles alone, without any cryopreservation agent, at concentrations up to 10 milligrams of iron per milliliter, we see no effect on cardiomyocyte viability. So we know that in this concentration range, the particles alone are not toxic to the cardiomyocytes. If we add VS55, the cryopreservation agent, we see that as we increase the amount of cryopreservation agent, there is toxicity. So this is what I mentioned before. 50% CPA has no toxicity, but by 75% CPA, we're killing a lot of cardiomyocytes. This is when we go from media directly to the CPA solution. If instead of doing that, we add the CPA solution gradually, basically avoiding any osmotic shock on the cells, we find that there is a reduction in cardiomyocyte viability, but it is a lesser reduction, and we can add the nanoparticles and retain about 75% of the cell viability. Next, we evaluate whole heart perfusion with these magnetic cryoprotecting agents. We remove the heart, we perfuse it with a cardioplegic solution called custodiol. Then we perfuse it with the magnetic cryopreservation agent, and we can do imaging at that point and histology. We can then remove it with custodiol and do more imaging and histology. And so these are images of a heart that's been perfused in with the cryopreservation agent. You see it's very dark because the particles are distributed throughout the heart. And this is an image for a heart that has been perfused in with the particles and then perfused out. So the particles have been removed, and you see that it goes back to its normal pink color. These are MPI images of these hearts. And so this is the control heart, no signal. This is the heart perfused in. You can see a fairly uniform signal throughout the heart. And this is the heart that's been perfused in and out. And so you can still see a faint signal. But if we quantify those signals, we see that the particles, about 95% of the particles have been removed. And this here is a rotating 3D image of those two hearts. H&E staining and histology does not show any difference in gross markers of damage. There's no really evidence of damage to the tissue in these cases. And the surgeon who helped us with this, Dr. Staples, he's a cardiac surgeon. And according to him, the feel of the heart was very similar to a fresh heart. You can see particles when we don't perfuse them out. You can use Prussian blue staining, and you can see particles dispersed in the tissue. But once you remove them, it's very difficult to see them. Finally, we demonstrate whole organ vitrification and nanowarming. And the way we do that is we remove the heart, perfuse it again with custodiol, perfuse in with CPA, then submerge in CPA and vitrify it using a mechanical freezer. We store them in liquid nitrogen temperature for 1 week, and then we nanowarm them using an alternating field. After that, we remove the particles we're using custodiol again, and we do iron quantification by MPI. And so here is an example of a successful vitrification and nanowarming. The top image is the heart loaded with the magnetic particles before being vitrified. This is the corresponding MPI. They're in different orientations. That's why they don't look the same. And this has about 3 milligrams of iodine inside the heart. This is now the same heart after being stored in liquid nitrogen for 1 week and perfused with custodiol to remove the particles. About 90% of the particles have been removed. The particle looks good. And so we had a couple of hearts where we were able to do this, but I think it's important to also acknowledge that this is still a new technology, and there's a lot of kinks that need to be worked out. And here it's interesting how MPI can also help us assess this. This is, for example, a heart where we had a left ventricle obstruction, and so you can see that about half of the heart has been perfused with the nanoparticles. It's dark, and another half is not. And that's also evident in the MPI. You can see the distribution of the particles in one half of the heart and not the other. This is an example of a heart that we were able to perfuse, but because of issues with the rewarming step, we did this one in the bath, so not in the magnetic field, so that we had thermomechanical stress. And so you can see evidence here of that thermomechanical stress causing damage to the heart. This is another heart where we didn't immerse it completely in the CPA solution before we preserved it. And so that meant that part of the heart was not as effectively protected from vitrification, from crystal formation. And so that caused damage. So despite these limitations, in some of the experiments, we're really excited because I think this is the first demonstration of successful perfusion, vitrification, storage, and rewarming, and then being able to remove all the particles. And we're hopeful that in the future we'll be able to demonstrate that these hearts are viable for transplantation. Before I finish, I'm going to highlight a few other examples of my research. We've also worked on formulating magnetic drug carriers that have MPI capabilities. These, for example, have a doxorubicin prodrug that hydrolyzes readily, but the prodrug is used to increase the hydrophobicity of doxorubicin, which seems counterintuitive, but really enables us to encapsulate them using flash nanoprecipitation at drug loadings that are as high as 50% by mass. So the particles are 50% by mass doxorubicin. And here TEM image is showing that this gray sphere is the precipitated doxorubicin, and the darker spheres inside are the iron oxide particles within. And those carriers are now MPI capable. And so we're interested in using these for image-guided drug delivery. Yesterday, I was asked about being able to eliminate or reduce the number of animals used. And so because of the pandemic, we had to limit our animal work. And so we pivoted to developing anatomically correct 3D printed phantoms that are suitable for MPI. And we did that by generating an anatomically correct computer-aided design model of mouse anatomy using the DigiMouse Atlas. And we turned this into a 3D printable and MPI, and we 3D printed it in an MPI transparent plastic. And for example, here I illustrate a situation where we have a liver cavity and a brain cavity. And what we can do is we can put known amounts of particles in each of those cavities and then take images. And this allows us to plan experiments under conditions that mimic the eventual animal experiment. It allows us to evaluate different imaging conditions. And I think most importantly, because we know precisely how many particles are in each region, we can take images, analyze the images with the way we would do with a live mouse, and validate our image analysis protocols. And so this, for example, a 3D printed mouse with particles in the liver and in the brain. The other three signals are fiducials of known mass that are used as internal calibration so that we can then quantify the signal in the regions of interest. And this is with 2D and 3D scans in high sensitivity mode. I'm showing in the x-axis the known amount of particles that we put in the brain, and the y-axis the amount of particles that were calculated using MPI. And the solid line means parity, means agreement. And we can see very good agreement over a fairly wide particle mass range in the 2D model, and fairly good agreement, some issues at higher masses, but fairly good agreement in the 3D model as well. Okay, so to conclude the tutorial yesterday and today, I hope I've conveyed that MPI is an exciting new biomedical imaging modality that can track superpower magnetic iron oxide nanoparticle tracers non-invasively, unambiguously, and quantitatively. Some of the highlights of our work that I presented is that stoichiometric addition of molecular oxygen during the thermal decomposition can be done safely. And it results in spions that have very uniform magnetic properties and enhanced heating and MPI performance. And then we can covalently graft PEG silenced to those particles to obtain MPI tracers with long blood circulation half-life. We can also leverage strong interactions in nanoparticle chains to obtain unprecedented MPI resolution and sensitivity. You can use MPI and the same concepts, physics of MPI signal generation to achieve image-guided and spatially controlled spion magnetic hyperthermia. I didn't talk about it today, but again, tomorrow you'll see it if you want. It's a pre-recorded lecture for tomorrow's conference. We can use MPI to track adoptive cell transfer T-cell cancer immunotherapies, non-invasively and quantitatively in solid tumors. And finally, colloidally stable spions can be formulated in cryopreservation agent solutions to uniformly perfuse whole organs, enabling their vitrification, storage, and rewarming, avoiding any damage from rewarming. And to close, basically, I think MPI is really poised to make an impact in tracking nanomedicines and cell therapies, and I hope you're as excited as I am. So again, let me thank my students, collaborators, and funding, and you for your attention, and I'll be happy to try to answer your questions.
Carlos, thanks for the excellent lecture. I have two questions here from people in YouTube. First, we are almost 10 minutes. We have 10 minutes to start the other lecture. So let's go as fast as we can with the questions. It's Dr. Rinaldi from Federal University of ABC. He has two questions here. He has a question concerning the linear chains data presented in the first part of the talk. Are them monodispersed chains? In case they are monodispersed, any idea what happens with non-monodispersed systems? This is the first question.
So in the oh, sorry, go ahead, Andres.
If they are not monodispersed, if you have any idea what would happen in non-monodispersed systems?
So in the models, the particles are monodispersed and the chains are monodispersed. There are only 10 particles per chain. Although we did look at the effective number of particles per chain and the effective particle size, the number of particles per chain affects the threshold field, but you still get the phenomenon. Anywhere, I think the lowest we did was 5 particles per chain and the highest was 30. Particle size affects the threshold field as well. In the experiments I showed, the particles are as monodispersed as you can make them, right? So there is polydispersity, and there's no control over how many particles are in each chain, and you still have the effect. And many of the features that I showed in the theory, the existence of a threshold field, etc., all of that translates to the experiments.
Okay. He has another question, but now regarding the nanowarming. Concerning nanowarming design criteria, have you considered aggregation and polydispersity?
So again, for the particles I showed, we don't see any evidence of aggregation based on dynamic light scattering. Polydispersity, in that application, I mean, if we wanted to get really good NPI images, we would want to get a particle that is more monodispersed. But heating from nanoparticles, in my opinion, is more forgiving to polydispersity than NPI. And so even though it's a co-precipitation particle and it has polydispersity, we still get sufficient heating. I mean, I showed you results with heating rates of 320 degrees C per minute. The heating rate needed is about 50 degrees per minute. Now, are we pursuing this with more monodispersed particles? Yes, because we would be able to get better NPI images.
So the other question comes from Isabella Ferreira. Good morning. Thank you for the great lecture. I would like to know, how do you measure the dose in the tumor do you use those emitters with nanoscale inside the tumor or just evaluate by the image?
Oh, say that again.
Sorry. She wants to know, do you use those emitters with nanoscale inside the tumor or just evaluate by the image? I would like to know how do you measure, she wants to know how do you measure the dose in the tumor?
The dose of particles or the...
Yeah, I guess the dose of the particles. You need to explain how they are getting the concentration with the NPI image signal.
Oh, okay. Yeah, so you
Yes.
can, in MPI, what you do is, again, the signal per voxel or pixel is going to be proportional to the mass of particles. And so we in a computer program, an image analysis computer program, this can be done in ImageJ, although we use 3D Slicer. You can define regions of interest, quantify the signal in that region of interest, and then you have another sample, a reference sample of the same particles at known mass, and you do the same analysis. And from the ratio of the intensities, you can calculate the mass in the tumor region. And you can do that in 2D, and you can also do that in 3D.
Okay. We have a question from Catherine Everert from PTB, German, in Berlin. The question is concerning the presentation of yesterday. No problem, right, Carlos? You explained that the response of the particle should be faster than the characteristic time scale of the driving field in order to limit the influence of the relaxation of the magnetic nanoparticles. Would it be possible to decrease the frequency of the applied field instead of decreasing the anisotropy constant of the nanoparticles to meet this condition?
Yeah, I mean, that's certainly possible. I know that there will be issues with the hardware. And what I mean by that is I'm going to do this by analogy with relaxometers, right? As you sample at higher frequencies, you get more flips of the dipole per unit time. And so you acquire more image, more signal. And so there will be limits to how low a frequency you can go and still be able to obtain a significantly strong signal that can then be quantified. But yeah, you can tune the frequency. And I want to point out that you would have to tune the frequency by order of magnitude. It's not enough to go from, so the momentum scanner operates at 45 kilohertz. So it's not enough to go from 45 kilohertz to 4 kilohertz. You would have to go from 45 kilohertz to 4.5 kilohertz for there to be a significant shift. And the reason for that is if you look at the frequency response of a magnetic particle, it has a very broad frequency response, and typically we look at it in logarithmic scale. Okay.
Okay. So Rony Mioto is thanking you very much for the talks and the answers. And I guess we are in time for the next lecture, Carlos. But I really appreciate a lot all your talks. It was very inspiring. The results are very nice. The nano warming, going to the nano warming is very cool. And Catherine is also thanking you for the answers. And we highly appreciate you in the second Advanced School of Nanomedicine. And for those that are not already registered in the workshop on nanomedicine and immunotherapy from the Brazilian Breast Cancer Symposium that Carlos just told about, that he would make that presentation there. That's happening now. I put here in the chat the details of how you can register for free there. Please go there. There will be several nice talks tomorrow during the Brazilian Breast Cancer Symposium in this workshop. It would start at 8 a.m. and going up to 6 p.m. Science is fun, so you have a lot of fun watching all those talks. Thanks, Carlos, again. It was really nice to have you here, and see you tomorrow.
See you. Thank you.
So, hello everybody. We are back here with the second advanced school on nanomedicine. We're now going to the last lecture of Dr. Robert Ivkov. Dr. Robert Ivkov, please.
Oh, you want me to begin?
Yes.
Okay, that was very quick. Okay, well, thank you. Thank you,
I'm
Andrew.
sorry.
My apologies. That was very, very fast. Okay, so let's see.
I had already presented you yesterday, Robert, just because of that.
Yeah, true. I'm just kind of, okay, can you see my?
Yes, yes.
You can see my screen, yes?
I can see your screen with several slides in the left too.
Yep.
If you put as a presentation, there you go. But let me see. Not yet. It's hide, and you need to do the presentation there.
I already did that. It is all showing on one screen. It's actually showing on my screens, but not on.
Yeah, it is not. It's showing your screen. It's not going to presentation, right? Presentation mode, Robert.
Well, I switched computers and I'm now on a third.
But we are able to see, we are able to see. I don't know if you're able to include the.
Well, so this is
If you pass please the slides so I can see how we are seeing.
Oh, it actually is not going because it is.
So you need to get out maybe.
Yeah.
There you go. It's moving for wants to the acknowledgements.
Okay,
Might pass.
Well, let's try this again. But so as you know, I had to switch screens. I switch my computer and everything at the last minute. And in doing so, I went to a different system that now has 3 screens. And so that, okay, I think what I'm going to have to do, unlike yesterday, is I am going to have to put myself into presentation mode first. And then
I think that, okay. You have the presentation is down below and below.
Yes. No, I sorry, I had to put my okay, so I think this will work. So I had to
Yes, it's working, Robert.
Okay. Yeah, so it's minor.
Slides. Is it passing slides? Yes, it's passing. Yeah,
okay.
It's nice.
All right, so apologies for that. We had some technical difficulties. Anyway, thank you again, Andres, for the invitation. I will go ahead and get started then. I will go through the sort of the background and a couple of example applications of magnetic nanoparticle, also called magnetic fluid hyperthermia. For this talk, I'll be calling it MHT or magnetic hyperthermia. And as before, as yesterday, I will say please feel free to send your questions to Professor Bakuzis, and Andres, please just go ahead and interrupt me with questions if that's okay. This slide is little change. There are a few additional or different names because some of the people involved in this work are different from the work that I showed yesterday. And let's see, I will put a laser pointer. Funding agencies also a little bit different because for this we've had funding from the Department of Defense, Prostate Cancer Research, NIH, as well as other donors. Again, the disclosures, financial disclosures, same as yesterday, and then without any additional introductory material, I guess we'll go ahead and get started. So The motivation generally for magnetic nanoparticle hyperthermia is really the same motivation as for generally for hyperthermia or thermal therapy for cancer, and that is that that heat or heat stress enhances the effects of other therapies such as ionizing radiation and some chemotherapies through complementary mechanisms. And this is something that is known. Many of these effects are known now for about 30 or 40 years. All of the effects or all of the details are not entirely completely understood, but well enough. The understanding has been well developed enough, and there are sufficient clinical trials data that really do emphasize the point that heat or heat stress enhances effects of ionizing radiation. And in fact, in many countries, hyperthermia is indicated for recurrent or refractory local recurrence of cancers such as breast cancer in combination with radiation therapy. And so the general, in a general sense, the effects of combining heat with ionizing radiation, biologically, the combination in the heat stress will inhibit DNA damage repair. As you probably know, ionizing radiation induces DNA double-strand breaks, DNA damage. And by combining, when heat is combined, then the repair mechanisms of the cells are either inhibited or abrogated, and the cells will die. Physiologically, the effect of heat on tissues is to increase perfusion, increase blood flow, and this increases oxygenation, which enhances radiation therapy. And we have to be careful here. Our medical physicists would probably make a note that this only works for low LET radiation, so photon based. It does not work for high LET. There is no oxygen enhancement ratio for high LET radiation, which is an interesting phenomenon in and of itself. And for those of you who are interested, there is an argument being made fairly much more recently by Dada and others that the relatively new modality of proton therapy combined with hyperthermia is a quote unquote poor man's carbon ion radiation. But that's getting into much more into the medical physics, so which we don't need to get into here. Heat or heat stress is also, people have argued that it enhances immunogenic cell death, and so therefore should enhance the effectiveness of cancer immunotherapies. And I'll touch on that in the talk today. So these things, these particular aspects of it are what I will touch on and present in a little bit more detail today. So the key advantages, at least a few of the key advantages that we consider to be offered by magnetic hyperthermia over other heat-based modalities, is that the nanoparticles provide a means to internalize the heat source into the tumor. So in other words, the heat can be generated inside the tumor, and the amount of heat that is being generated, which is a magnetic loss power, which is a property of the physics, it can be adjusted remotely by adjusting the power or the amplitude of the magnetic field device. So the nice thing that we consider here is that key advantages of magnetic hyperthermia is that the nanoparticles, being the heat sources, can be injected directly into the tumor, and then that the heating can then be remotely controlled. And a particular advantage of this over other modalities, for example, laser-induced or other modalities, is that tissue does not attenuate magnetic fields. So in principle, it is possible to treat tissues deep in the body, tumors deep in the body. And then one of the things that we actually take advantage of to overcome, or at least to address some of the challenges of magnetic hyperthermia, is that there is a thermoregulatory response. and that this regulatory response is plastic and dynamic. In other words, tissue that is heated, which of course, if you remember up here, this was one of the advantages of heating, but tissue that is heated will respond to mild heating or mild heat stress by increased vasodilation. And this increased vasodilation increases blood perfusion, which now changes the thermal properties, the dynamic thermal properties of the tissue. And we actually take advantage of this in some of the work that we do. And I'll touch on those aspects a little bit in this. Nanoparticles as a potential cancer immunotherapy. And so obviously I won't spend any time on that in this talk today. So the concept of magnetic hyperthermia is really illustrated in this cartoon here, and that is we have a nanoparticle in suspension in a biocompatible fluid, suspended in a biocompatible fluid, water or sterile saline, and that these are then injected or somehow delivered to the tumor, either directly injected into the tumor or, as in my talk yesterday, was intravenous injection. For everything I'm going to talk about today, I will be focusing on direct intratumor injections of the nanoparticles. So please do not confuse the material from yesterday. Yesterday was all about systemic exposure. Today is focused on hyperthermia. Everything that I'm going to be talking about is local exposure, so local injection of nanoparticles into the tumor, and then a focused treatment of heating and radiation therapy. And then we apply an alternating magnetic field, and through physical property, I'll show you some data on this, but through physical property called magnetic hysteresis or hysteresis loss power, the nanoparticles heat and they deposit their heat into the tumor, which then sensitizes the cancer cells in the tumor to either radiation or to drug, or will kill the cells outright if the temperature is high enough. And something that many people are either unaware of or forget is that magnetic hyperthermia, MHT, is actually a clinical modality. It was approved in 2010. The company that has that offers this is Magforce AG. They're a German company, so this was first approved in Germany, for use in Germany for treating glioblastoma, brain cancer. And the company is running clinical trials for prostate cancer and on glioblastoma in the United States. Okay, so we're going to step back a little bit now and walk through some of the details a little bit for those people who are interested in the physics. There is actually really quite a lot of interesting physics here that we can talk about, and I will only give you a sort of a summary of this. Professor Bakuzis and I spend a great deal of time talking about this. We have a lot of discussions on the physics. But what is interesting and what this graph shows you is that the amount of heat and actually the dependence of heating properties of the nanoparticles in response to the magnetic field amplitude is really actually very different and highly variable, and depends very much on the nanoparticles and their magnetic properties. And so one of the ways that we do this characterization is what we call the specific loss power, and it's a measurement. It's basically derived from calorimetry, and the units we give are in watts per gram of iron. We are working with iron oxide-based nanoparticles. The iron is the active ingredient, if you will, being the magnetic component. And so we, excuse me, we normalize our SLP measurements or the watts per gram to the mass of iron. And then on the x-axis here, you have in units of kiloamps per meter is the magnetic field amplitude or the field strength. So one of the things that I always like to point out to people is this material here, which is Faradex. It is a super paramagnetic iron oxide. It does not heat. Superparamagnetic materials, superparamagnetic iron oxides, or magnetic nanoparticles that exhibit superparamagnetic properties are not going to heat in an alternating magnetic field. There is another material that we actually published on this. These are the materials that I talked about yesterday that we used for the biodistribution. Even though they're called SPIOs, clearly they're producing some heat. So some portion of these nanoparticles is not actually in a superparamagnetic, is not exhibiting superparamagnetic properties at the conditions that we do the experiments. So they do exhibit some heating, but it's actually fairly weak. And so there's actually a fair amount of work, and I was involved in developing both the BNF and the JHU magnetic iron oxide nanoparticles. And you can see that the heating properties of these two are actually very different. And it's these differences that actually created a fair amount of interest for me on the physics. And I've, as one of the things that I've been working on for probably more than a decade now, is really digging into and characterizing some of the nature of the nanoparticles, of their magnetic properties that gives rise to this difference. And then here's a series of publications on this. So here's a cartoon, and this is basically a pictorial representation of what we call hysteresis. And so what we're talking about, if we consider the nanoparticle to be a magnet, and if it is put into, if it is exposed to a magnetic field, many of you might have actually done this as kids, where you take a material that is potentially magnetic, like a nail, and you put it on a magnet, it will magnetize. That's what we would always call it. So if I take a magnetic material and expose it to a magnetic field, then I can measure a magnetization in the material. In other words, the material becomes magnetized. And as the strength of the external field increases, so does the magnetization of my material. And now if I reverse the magnetic field and go back down to zero, start taking away the magnet, what I can find is that in a ferro or ferrimagnetic material, I will see a residual or remnants magnetization. In other words, the material is itself now a magnet. It has memory. And so now if I continue to force that magnetic material or the magnetic moment magnetization down, I will eventually cross the zero again. And then, excuse me, if I continue in this direction, I will essentially generate a path, a magnetization path that goes out to the saturation on the opposite side, so the opposite direction. And then if I repeat that process, then I'm going to retrace, but now a different path. So a ferro or ferrimagnetic material generates what we call an irreversible path. And this is a key feature of, or a key requirement of a material to be able to generate heat with an exposed magnetic field. Things get very complicated when we have individual nanoparticles that are suspended in a fluid, because depending on the orientation of their crystal axis, that hysteresis loop can present different area to the magnetic field. And so now we can have a distribution of energy responses in our magnetic nanoparticle suspension. And so now we get into a situation where things get much more complicated because of all of these variations, and because the nanoparticles can reorient and can move, and they also interact with each other. So there is a potential for dipole interactions, because magnets are, magnetic force is a long-range force. And so now this magnet may actually influence this magnet, and so it might change the hysteresis behavior, hysteresis properties. So there's orientation, there's collective or interparticle interactions, and all of these effects average out in a suspension of nanoparticles to give us a really complex average or mean effect. And if you recall, I told you that one of the things that has interested me on the physics of these things and applied physics is really what is going on inside the nanoparticles that generates these very different heating curves. And so I won't get into this because that's an entirely different talk, and we published this work in Advanced Functional Materials in 2015. And by the way, this was a great deal of work. It was about 6 or 7 years worth of work with collaborators. But what we basically concluded from this study is that all three of these particles are essentially from very sort of large scale or bulk measurements. They all have very similar features in the sense that their bulk magnetization saturation, their diameter, and all of these things were really not very different from one from the other, and yet they had these very different, they exhibited these very different responses to an alternating magnetic field, the amplitude dependence of an alternating magnetic field. And we used polarization to analyze small angle neutron scattering and some other techniques to really look at the internal magnetic and structural features of these nanoparticles. And what we essentially concluded is that because of the way the individual iron oxide crystallites are arranged in these materials, they actually generate a collective magnetic state that gives very, very different energies and very different responses to an alternating magnetic field. And the only thing that is I want to sort of emphasize here, and unfortunately this might be a small figure, but basically this number, kiloamps per meter, 20 at the frequencies we're using, 20 is probably about the limit that you can go for treating humans. So really a big an important driving feature of this entire development was to find ways to make nanoparticles that are much more responsive to the magnetic energies at the low end so that we could actually have a clinically viable nanoparticle. I think earlier you did hear from Professor Rinaldi both yesterday and today talking about magnetic particle imaging and its combination with magnetic fluid hyperthermia, or MHT. And again, just to repeat, magnetic hyperthermia has been clinical modality since 2010. It does offer some advantages, which I touched on, but the lack of precise imaging, and this is one of the aspects that I'm going to dig into a little bit more in more detail, and control of the nanoparticle activation will create a need for technological solutions to essentially enable a wider clinical implementation. And I'll get into the details on that. That's sort of my problem statement for today's talk. And what we're really going to be talking about is what are the key challenges with the nanoparticle heating or magnetic hyperthermia that we have to deal with. So one, there is actually a challenge to depositing the energy in a controlled way so that one achieves a temperature control that can give you towards a prescriptive treatment plan that is that compensates or that can overcome the heterogeneous nanoparticle distribution inside the tissue. And then there's also off-target tissue heating that can arise from nanoparticles that are outside the tumor and also from interactions of the magnetic field with tissue. There is actually a challenge in that the standard imaging technologies do not image iron oxide nanoparticles very well at the concentrations of nanoparticles that are needed inside the tissue. So we don't have the needed spatial resolution and the precision to be able to perform really accurate treatment planning. And then finally, there is also a need to develop a volumetric thermometry that can give us a much more accurate picture of the temperature distribution throughout the tumor so that we can perform quality assurance of the treatment to make sure that when the treatment is concluded, that the treatment actually conform to the treatment plan and how much or how accurately it conformed. And that really is one of the when I would argue that that's one of the challenges that is presenting a barrier to wider clinical implementation of magnetic MHT today, is really that that the current modality does not offer sufficient quality assurance and quality control of energy and temperature, energy deposition to achieve temperature control to conform to a treatment plan, and then to provide the volumetric thermometry to establish quantitative quality assurance that the treatment plan was executed. And this is where magnetic particle imaging offers that potential. So that's really all I'm going to say on the introductory material. Now we'll just sort of focus in on some of the more detailed aspects of magnetic nanoparticle hyperthermia, or MHT, and in particular with radiation as a radiation sensitizer. So this, I think this figure, I think, is a really good illustration of one of the principal challenges with the magnetic nanoparticle hyperthermia, or MHT, even with direct intratumor injection of nanoparticles. So what you see represented here are whole tumor tissue sections from human prostate cancer xenografts. They were subcutaneously implanted into nude mice. So remember the discussion yesterday about the nature of the animal models. This is an example where I think our nude mouse models are perfectly fine for doing the work. And what you see here is basically all of these tumors were injected with nanoparticles in as best as one could do. The amount of material that was injected, or the concentration, was the same in all three cases: PC3, Du145, LAPC4. These are the different cell lines from which the tumors are derived. And you can see that in the case of PC3, there is really essentially a fairly sort of widespread nanoparticle diffusion to the edge, one particular edge or boundary of the tumor. In the case of Du145, we really, from what we can conclude, we probably lost a lot of the nanoparticles, and actually our ICP mass spectrometry confirmed this. LAPC4, interestingly enough, gave us a really, actually, what I would call an ideal distribution for nanoparticle hyperthermia, in the sense that we have nanoparticles that are actually, we have aggregates of nanoparticles that are distributed more or less throughout the tumor. And these tissues are stained with Prussian blue, so the blue staining really does give a good indication of the nanoparticle localization. Yes, sir.
Robert, we have a question. I just would like to remind you that the other talk would start at 11 o'clock, just for you to keep that in mind. But it's coming from Edinara, from Federal University of Goiás. She has the following question: thermotherapy takes advantage of the fragility of cancer cells, but at the same time, the cells are considered to be in constant evolution. I would like to know if cancer cells can become resistant to thermotherapy, just as happens in conventional therapies. Can thermotherapy replace conventional therapies, or just be used as a complementary way?
Yeah, very, very good question. So that's not just one question. There are several questions. But but they're all actually very good questions, key questions. So answer number one: there is actually inherently no cancer cells in general do not exhibit a greater sensitivity to heat than normal cells. There is enough variation among normal cells in your body in the sensitivity to heat that basically cancer cells don't actually show anything, as a general rule, don't show anything different from that. The differences really come from, there is a difference in that tumors show greater sensitivity to heat. And I mentioned that it's because the mildly heated tissue will show increased perfusion. Increased perfusion leads to greater oxygenation. This doesn't really help thermal therapy so much, but it actually helps radiation. And to the final question, absolutely correct. Actually, there is another one. Yes, cells do develop resistance to thermal therapy. So non-lethal a non-lethal exposure to heat stress will create a response in cells. Cellular mechanisms will respond to increase their heat stress response, heat shock proteins. And what you find is that cells that are exposed to heat, that are to non-lethal heating, will show resistance to the same thermal dose if it is applied again, which lasts typically about a week. And this does actually have a clinical implication. Mark Dewhurst did some work in the clinical trial in canine patients, sarcomas, and actually demonstrated that fractionated hyperthermia, if the fractions were less than 1 week apart, did not actually improve or confer an additional response. But if you wait a week, then that sensitivity is recovered, or in other words, the resistance to the heat is lost, and now you can recover some of that, a great deal of that sensitivity to heat. As a general rule, hyperthermia is not more potent as a single agent cancer therapeutic than is either radiation or chemotherapy. So clinically, in most countries where hyperthermia is approved for use, it's approved as a treatment modality to be combined with radiation or chemo.
There is another question, Robert, from Rohan from Federal University of ABC. When you say that the third image is the ideal distribution, since aggregates are seen throughout the tumor, you imply that aggregates are essential to magnetic hyperthermia or only improves its effectivity?
So the short answer is yes. And so yes, if a person, for those of you who are immersed in the heat transfer, basically so just as a very sort of short response to that, and I'll show you some data with this next slide, but the short response is that the aggregated nanoparticles or aggregates of nanoparticles are going to generate higher heat, and so their rate of heat loss or heat transfer outside of that area is essentially such that the nanoparticles that are individual or homogeneously distributed, the rate of heat loss from these is such that the heat transfer at the tumor tissue boundary is greater, and so it's more difficult to sustain you have to sort of apply a bit more energy to sustain a thermal temperature gradient inside the tumor relative to the normal tissue. So what happens is basically when you have to take into account heat transfer and the heat transfer properties of tissue, particularly with blood flow and perfusion, what ends up happening is that the uniform distribution of nanoparticles is not really as ideal as some localized concentrated or localized aggregates of nanoparticles, because now they can generate a great deal of heat, and then you can let that heat transfer out of the tumor. Of course, that now implies that one has to use some different strategies for managing that heating. So then that question actually leads into this slide. So one of the things that we did in order to actually look at exactly that question, what's sort of the ideal distributions of nanoparticles and how do we compensate for heterogeneity of the nanoparticle distributions? And again, one needs to remember that whether we directly inject the nanoparticles into the tumor or even systemically administer the nanoparticles to the patient or to the animal and then have the nanoparticles being directed to the tumor, you're never going to see this idealized uniform distribution. It's just not going to happen. Even this tends to be a rather difficult to achieve. We assumed that by directly injecting the nanoparticles into the tumors, that we would see something more or less like this, and instead we saw this. And so really one has to acknowledge and accept the reality that the nanoparticle distribution is out of one's control after injection, and where the nanoparticles are going to end up in the tissue is what you've got to deal with. You're not going to get what you wish for. Okay, so this is where we do the heat transfer, bioheat transfer simulations. And this is just to give you, and again, we published this work, but basically you can see, so here what we did was we applied, if we considered in our simulation, we considered each pixel. So going back up here, okay. My apologies for, I pressed the wrong button. So going back up to this, what we did was we digitized the histology images, and then in the digitized samples, we treated each of these pixels as a nanoparticle or as an independent heater. And then we applied a heat term or a Q, an energy term. And so now we let the simulation run. And what you can see is that the PC3 with the nanoparticles distributed in that area, they produce a generally concentrated heating that then distributes throughout the tumor. The DU-145 was off-center. And now you look at the LAPC4 and you see that the LAPC4 type, which is this, gave us an interesting difference between this reality and the uniform, the homogeneous nanoparticle distribution, in the sense that we can actually achieve a higher temperature inside the tumor. And one of the things that we also noticed is that it was actually, it may not be as well represented here, but what we found, which is equally important, was that at the tumor tissue boundary, we could limit the damage to the normal tissue better than we could in the case of the uniform distribution. So one of the things that strategies that we decided to implement, and it was, if we have this kind of a scenario, and up here we have a constant power. So in this case, in the simulation, we just turn on the power and we leave it as a constant power. So our strategy to compensate for this distribution, or at least partially compensate, is what if we adjusted the power to modulate the heating, where we use temperature feedback control. And so this exercise is a series of simulations where we say, okay, where is the ideal placement of the temperature probe? Is it at the center? Is it intermediate between the center, halfway between the center and the tumor tissue boundary, or do we place it at the tumor tissue boundary? And then do we adjust, we have some criteria for the heating where we adjust the power to achieve or to maintain some temperature, some target temperature at that probe location. And as it turns out, as you can imagine, putting the temperature probe at the tumor tissue boundary is actually the best place, because what we are trying to do is achieve as much heating inside the tumor, but minimize the damage outside the tumor. And so our criteria were actually twofold, where we had two different constraints. And it turns out that putting the temperature probe at the tumor tissue boundary, if one has only a single temperature probe, that this is really sort of the best scenario. So implementing this type of a strategy in our mouse models, we ended up with this. So what you're looking at here is actual temperature measurements from a mouse that was bearing one of these tumors, either PC3 or LAPC4. And so here are temperature readings from a temperature probe that was inserted inside the tumor. The black solid is from a location opposite the tumor in the skin. And then we also measured the rectal temperature of the mouse, because what we want to do is we want to monitor and make sure that the mouse temperature, or the mouse, doesn't overheat, and talk about that in a little bit more detail a little bit later. So here's our constant power modality. As you see, basically the temperature keeps ramping up and get to a very high temperature until the end of the treatment, when we turn off the power. And here we implemented a strategy where we had a fairly high amplitude at the beginning to achieve a quick heating, and then we started to manage and control the temperature and really basically achieve or control the tumor temperature to a target of of about 43 degrees C, which is the mild hyperthermia threshold. And so now when we look at what happens, so after treating several mice with this and combining it with radiation, what I didn't also show you is that PC3 is actually relatively resistant to either heat or radiation combined. They do induce more cell death. LAPC4 is inherently more sensitive to either radiation or to heat. And we can look at the Kaplan-Meier here, which is essentially a measure of an aggregate measure of the tumor growth. So our no-treat controls, basically, the tumors reach 4 times their initial volume within about 22, 23 days. The red line is the hyperthermia. All of these are a single treatment, a single fraction of heating. And here we are doing the 43 degrees C heating. And then here we compare with 5 gray of radiation, single fraction, single agent. And then in the case of the LAPC4, we combine the heat, the MHT with the 5 gray of radiation, in which you see there's a significant improvement in the response. In fact, it's even better than a single dose of higher, a single fraction of higher dose radiation of 8 gray. The 5 means 5 gray, 8 means 8 gray. PC3, we see a very similar scenario in the sense that the combination does actually improve from either one agent alone. But in the PC3, it's a more challenging model. And so we don't see that there is a greater response than the higher dose of radiation. So at least, in this model, what I could basically show you was that the combination of MHT, when it is applied in a way where we modulate the power to compensate for the nanoparticle distribution and combine it with radiation, we do actually achieve a locally improved control. And so the last thing that I want to show you is how we induce abscopal effects combining radiation therapy with the magnetic hyperthermia and immune checkpoint inhibitor therapies. And actually, a more detailed version of this portion is going to be in the workshop tomorrow at the conference. But for those of you who are not planning to attend that, I'll just give a little sort of a brief summary of this. So in this particular case, if you have seen the talk from yesterday, you might recall this model, the 4T1. This is a highly aggressive metastatic breast cancer, triple negative metastatic breast cancer model. It is a syngeneic model. So here we are talking about a mouse-derived cell line that where the cells are implanted into a mammary fat pad. So it is an orthotopic tumor. It's in the organ. So the cells are injected 7 days before we do our treatments. We have systemic immune checkpoint inhibitors, so anti-CTLA4 and anti-PD1. And then on day 0, the mice are treated with local hyperthermia, some combination of local hyperthermia and local radiation. Radiation is 3 fractions of 8 gray daily. And then one group of mice was sacrificed at 7 days and another at 14 days. Again, this is a very highly aggressive model. So within 14 days or 28 days after implanting the cells, basically we have very large tumors, and we also can find advanced metastatic disease or metastases in the lungs. The treatments, local hyperthermia or MHT treatments, were mild hyperthermia, so 43 degrees C for 20 minutes, using the methodology that I already explained. We also tried a partially ablative, where we went up to 45 degrees C for 5 minutes and then dropped the temperature down to 43 for 15 minutes. And then the radiation, and then the immune checkpoint inhibitor, and then various combinations of those. So one of the things I want to highlight is I'm a big fan of including controls. And very often in these abscopal or immunotherapy papers, you don't see this control. This is a surgically resected control. So in other words, this control is the mice with no treatment. Here, what we did was at day 0, the day that these other treatments were taking place, one group of mice, we did nothing other than to just remove the primary tumor. And that gave us some very interesting results for comparison. So you see there's no other treatment here, just removing the tumor by surgery. All right, so then these are all color-coded. So we have our single treatments. This is immunotherapy. This is the mild hyperthermia. This is the ablative hyperthermia. And then there's radiation. So what we're looking at here is the tumor volume of the individual mice. So this is a box whisker plot. Tumor volumes, measured tumor volumes at day 14 at the endpoint of the tumors in the individual mice. So this is primary tumor with these different treatment combinations. So now here we have the dual treatments. So now here we have mild hyperthermia with immune checkpoint inhibitors. We have ablative with immune checkpoint inhibitors. And then we have radiation plus immune checkpoint inhibitor and radiation plus mild hyperthermia, radiation plus partially ablative. And now we have the triple combinations where here it's all 3 with mild hyperthermia, and here it's all 3 with the partially ablative. So what you can see is that the dual combinations and the triple combinations give you much better control than any of the single agents, particularly when radiation is combined. And if we were to make an argument for any single agent, it would probably, the winner would have to be radiation. Radiation still is our best single agent. But notice that surgery is surgical resection is giving us a result that is about equal to all of these treatments. What we did with these mice as well was we did histopathology analysis and did immunohistochemistry to stain for CD3. CD3 is a marker for T cells. So we're not distinguishing here between CD4 or CD8 T cells, but basically all T cells in the primary tumor, and using the same sort of scheme for looking at the treatment code, which you can also see down here. You can see that as we increase the combination treatments, we basically are seeing an increased infiltration of T cells at the endpoint. And so what this tells you is that these combination therapies are more immunogenic. They are actually causing a more stimulation of the adaptive immune system in response to the treatment. And I think that's very interesting. And to basically put a finer point on this, what we did here in this experiment, so it's the same sort of an experiment, but now some of the mice had, some of these groups, we added a blocking agent to T cells. So in other words, we killed the T cells with a pharmacologic agent. And what you see that in the case of the triple combination, we do see that a significant amount of the tumor response is due to activated T cells or activated immune response, because when we kill off the T cells, we see a lot more of that going away. We don't see that for these other treatments. And I think that's an important piece. So the conclusion that we can come from this is that adding the mild hyperthermia or adding MHT increases the response of the tumors or increases the effectiveness of the therapy, radiation and immune checkpoint inhibitor therapies, because it increases the adaptive immune response to the tumor. And I think that's a very interesting conclusion. One of the things so this is now we say what happens to the metastases or the metastatic tumors in the lungs. And here we're looking at measurements of the numbers from histology, numbers of metastatic foci in lungs as a function of all of the treatments. And one of the things that is really important to note is if we did not have this surgical resected control, we would be writing a nice paper showing how we had massive abscopal effects and very significant responses. But when we include our surgical resected control, we actually are finding that our effect is relatively more modest. So in other words, one conclusion from this is that with a single fraction of hyperthermia, combined with 3 fractions of 8 gray radiation and dual checkpoint inhibitor therapies, we actually achieve a control of metastases that is actually similar to surgically removing the tumor at the same time. And I think that's important because in this case, we are not yet able to distinguish between reduction in metastases of simply killing off the primary tumor or killing off cancer cells in the primary tumor versus the immunologic effect. Now, we know that we have a significant immune effect, an adaptive immune response in the primary tumor, which is enhancing the effectiveness of the treatment in controlling that primary tumor. What we cannot distinguish at this point is how much of the control of the metastases is due to that immune-mediated abscopal effect, or to simply cytocidal or reduction of the tumor. And one of the things that we really think that we can improve on a great deal is to enhance the effectiveness or enhance survival by taking this MHT, the mild hyperthermia, which did actually show quite a bit of promise, quite a bit of benefit. And now here we, coming back to that original question, we think that what we want to do here is include more fractions of the hyperthermia with the radiation. But if I was able to convince you, one of the things that we also understand is that our treatment is probably not quite getting all of the tumor, because our quality assurance of the effect of the extent of treatment or the degree to which we are able to treat is not quite there yet. And so that's one of the challenges, and this is, I think, a consequence of the challenges that we face with MHT. Even with directly injecting the nanoparticles into the tumors, we need to find ways to get a much better control of the energy deposition in the tumor, the distribution of the energy within the tumor, so that we can perform our treatments to a plan, and then have measurements, thermometry measurements, that can give us the assurance of our treatment to that plan. And with that, I'm going to that's my summary and my conclusions. In the interest of time, I am going to end.
Robert, thanks for the talk, for the excellent talk. We have some questions here. I would pass to the question of Honey. Robert, yesterday you have shown us another work with a similar conclusion: stimulation of the immune system by nanoparticles. What are the differences between these nanoparticles and the ones from yesterday?
That's a great question. So the difference between the nanoparticles yesterday and these today, there is none. They're the same nanoparticles. The difference is, however, that the nanoparticles yesterday were simply just the nanoparticles being intravenously injected at a fairly high dose, and we didn't do anything else. Here today, what I showed you was directly injecting the nanoparticles into the tumor and then activating them to generate heat, hysteresis heating with a magnetic field. So those are the differences. But René is correct. I mean, you can see that where we're going with this is that we think that might be a potential to combine the systemic with the local magnetic nanoparticle therapies, where the systemic is more of an immune modulating effect and there's no hyperthermia, with the local hyperthermia activation to increase the effectiveness. That is actually technologically much more challenging without being able to confine the magnetic field, which is actually the going back to the talk of Professor Rinaldi, where MPI guided MHT. Our current technology, we don't have that fine a control to limit the heating to only the tumor, because we we end up with most of the nanoparticles in the liver, and we know that we damage the liver significantly if we apply heat throughout.
Robert, we're almost out of time. I would like just to make a question from myself is, do you think that your control, your surgery control, certainly it should depend on the time, right? Depending on the time that you choose, this might be different. I mean, the comparison. So why did you choose, what was the size of the tumor that you had chosen as day 0? And why did you choose this time instead of the others in relation? Certainly it was related to the metastasis probably that you were checking. Can you make a comment on that, please?
Yes, no, that's a very good question. So first of all, the surgical resection was at the same time relative to implanting tumors as was the magnetic hyperthermia and the radiation. Okay, so 7 days, if you bear with me, 7 days before, so we had at day -7, the tumors were all implanted. So a certain number of cells was implanted into the mammary gland of the mice. And then at day 0, so it was a timed point. At day 0, all mice received were put into at randomly assigned to one of the groups and either received no treatment, surgical resection, or one of these treatments.
Hey, Robert, thanks. Thanks, Robert, for answering. We are just very busy here. Rohan, thank you very much. Robert, very nice talk.
Thank you.
And Robert, we need to start the other presentation. I know that other questions would appear, but you can send to Robert in the near future. Robert, thanks again for participating.
Thank you very much. It was enjoyable. So.
Yeah. Thanks for participating in the second Advanced School of Nanomedicine, and see you tomorrow in the workshop on nanomedicine and immunotherapy.
Yes, thank you. Okay, bye.
Bye, Robert. Bye. Max, pode começar. Hello, everybody. We are back again to the second.
Nanoparticle-based photothermal therapy generates tumor cell killing. In lecture 2, on the second part, we talked about how photothermal therapy, or PTT, activates the immune system. We touched upon the concepts of immunogenic cell death and how that can be measured. We talked about how PTT can cause dendritic cell maturation and secretion of pro-inflammatory cytokines. And we also talked about improved T cell phenotype and function. But we also discussed how PTT alone is not sufficient, because while you do potentiate an anti-tumor immune response, there are concomitant upregulation of immunosuppressive pathways, and as a result of which PTT cannot tackle dissemination. I presented the example of where Tregs infiltrate into the tumor microenvironment of an untreated tumor after PTT. There's also an upregulation of immune checkpoints, and other studies have shown that regulators or suppressor cells, such as myeloid-derived suppressor cells, infiltrate into the tumor microenvironment, thus damping the effect of PTT. So our motivation is to combine photothermal therapy with immunotherapy to see if this tandem approach can result in better treatment outcomes. So I wanted to give a brief overview of immunotherapy for those that are uninitiated. Any treatment that interacts with the immune system is classified as an immunotherapy. So why are people interested in immunotherapy? It is because when using immunotherapy, it offers the potential for robust and persistent treatment responses that may not be observed with other treatment modalities. In fact, it was recognized as such an important breakthrough that it was featured in the Science magazine now almost 8 years ago. But since then, there has been explosion in the development of the field of immunotherapy. So with the recognition that immunotherapy is important, the Nobel Prize for Medicine in 2018 went to 2 scientists that worked on immune checkpoint inhibitors, Dr. James Allison, who was then at the University of Texas, and Dr. Tasuku Honjo, who was at the University of Kyoto in Japan. So what we're really hoping to do is to use photothermal therapy plus immunotherapy to generate an abscopal effect. Now, it is important to define what I mean about what I'm talking about when I just use the word abscopal effect. The abscopal effect is a phenomenon wherein the benefits of a localized therapy extend beyond its treatment zone. Now, this was observed in the 1950s with radiation therapy, where they were able to radiate an individual tumor, and that treatment resulted in the shrinkage of non-treated tumors. Now, subsequently, the understanding of what players were important in generating an abscopal effect was developed, and it was then determined that the immune system plays a very important role in driving an abscopal effect. So, ideally speaking, we would want our PTT to be delivered into the tumor microenvironment, and then in combination with immunotherapy, trigger a cascade of events that will generate an anti-tumor immune response that can take care of non-PTT treated tumor metastases. There are several subjects available, good reviews are available on this subject, and I encourage you to read the most up-to-date literature on the abscopal effect. So briefly, what are the classes of immunotherapy that people are interested in? We have the immune checkpoint inhibitors, anti-CTLA4, anti-PD1, anti-PD-L1 that have received approval, and several others that are in development. There are adoptive cell therapies such as tumor-infiltrating lymphocytes, CAR T cells, natural killer cells, and other immune cell populations, including macrophages. You could have monoclonal antibodies, wherein antibodies are unconjugated or conjugated with a drug known as antibody-drug conjugates, or design antibodies to be bispecific. There is an FDA-approved oncolytic virus that comes under the umbrella of immunotherapy. There are also cancer vaccines, such as Gardasil for HPV. And there are other immune system modulators, such as interleukins, interferons, and immunomodulators. For the purpose of today's talk, I'm going to focus on the use of immune checkpoint inhibitors and immune system modulators. So immune checkpoint inhibitors, what is it that they do? They reverse immune suppression in the tumor microenvironment by targeting key immune checkpoints such as PD1, PD-L1, and CTLA4 in the nexus between tumor cells and T cells. By targeting these immune checkpoints and reversing immune suppression, what is observed is we see durable and sustained survival benefits by targeting these checkpoints. Now, this response of durable and sustained survival benefits have been observed in a variety of treatment tumor types, including melanoma, non-small cell lung cancer, kidney cancer, bladder cancer, and Hodgkin's lymphoma. And this is an over-expanding list of potential tumor targets that can be targeted treating using immune checkpoint inhibitors. Now, that's the promise. What are the challenges of working with immune checkpoint inhibitors? One key challenge is that you could have modest response rates in some of the tumors that are treated with immune checkpoint inhibitors. In some cases, the response rates can be as high as 50 to 60%, or in some tumor types, there is no response at all. In addition, you have immune-related adverse events, grade 3 and grade 4 adverse events. And this immune-related toxicity can be quite debilitating to the patient. So our hope is to combine nanoparticle-based photothermal therapy with immune checkpoint inhibitors to improve the response rates and potentially decrease adverse events. So how are we doing that? We use Prussian blue nanoparticles as the workhorse platform in our research group. So many of you have seen this iconic painting of the wave by Hopkins. The blue in this painting is made up of indigo, a small molecule, and Prussian blue, a colloidal nanoparticle. So now why are we interested in working with Prussian blue? For one, it is already FDA approved for radioactive poisoning, marketed as radiogardase, and it's on the WHO list of essential medicines. They are easily synthesized, these nanoparticles, at extremely low costs. And so that has implications for scalability and rapid manufacture of these nanoparticles. This was a concept I touched upon in lecture 1. And more importantly, the way the nanoparticles are synthesized, they can biodegrade into their constituent ions and be eliminated over time. And this provides some conferred advantages over some other platforms of nanoparticles being considered for PTT. So in terms of the workflow, how are we working with Prussian blue nanoparticles in this nanoimmunotherapy format? We first synthesize the nanoparticles. For this, we use a one-pot synthesis scheme. It is merely a combination of the 2 liquid solutions in a single pot to synthesize the nanoparticles. After we synthesize the nanoparticles, we isolate them, and then we characterize them for their size distributions, their surface charges, their short and long-term stability, and other signature properties. We also use techniques such as dynamic light scattering, zetametry, absorbance spectroscopy, and electron microscopy. Then, depending on our end use, we may functionalize these Prussian blue nanoparticles, either with targeting ligands, antibodies, adjuvants, or any other additive agent. And we use a whole host of conjugation methods, such as adsorption, layer by layer of opposing charged materials, specific interactions, and/or covalent interactions. And then after we synthesize, characterize, and functionalize the nanoparticles, we test them in vitro using target cells as well as normal cells, and in vivo in animal models. We have tried a whole host of animal models. I'm going to present some of that data today, but they are mainly syngeneic and some of them are xenogenic. And after the in vivo studies, we do ex vivo analysis. This is the post-PTT nanoimmunotherapy analysis to see any correlative effects of the therapy or toxic effects post-treatment. So how are we using the Prussian blue nanoparticles for photothermal therapy? As you can see here, this is the chemical structure of Prussian blue, soluble Prussian blue, if you will. And this is the absorption band on the right hand, showing a near-infrared absorption band. Now, we synthesize these stable Prussian blue nanoparticles, and we intratumorally inject them into a tumor microenvironment. We choose to do this intratumorally because if we systemically administer the nanoparticles, we would have to rely on the enhanced permeability and retention effect to get a sufficient dose of the nanoparticles into the tumor. And it has been shown that while it works in animal models, this is not necessarily translatable to human tumors. And so we inject them into the tumor microenvironment. So the question is, how do we do it translationally? This can be done through interventional approaches where catheters can be used to administer the nanoparticles to the locations where we want them to be. And then when we irradiate these nanoparticles with a near-infrared laser, so we use a laser of wavelengths 808, although that does not correspond to the maximum absorption of Prussian blue nanoparticles. It does correspond to the window of lower absorption of human tissue. And with this nanoparticles in the tumor microenvironment irradiated with the laser, we generate tumor cell death. Now, I talked about this in lecture 1, but we are not only interested in tumor cell death, we are very interested in the release of antigens and endogenous adjuvants from the tumor cells. These endogenous adjuvants signal to the immune system and there are many of them, including damage-associated molecular patterns. And what we're really hoping to achieve is that this localized intervention potentiates a robust systemic anti-tumor immune effect, which in combination with immune checkpoint inhibitors can maximize the abscopal benefit of our nanoimmunotherapy. So I've used the word nanoimmunotherapy. What do I mean by that? So it comprises of two components in its simplest form. It comprises of the Prussian blue nanoparticles used for PTT, and it consists of the immune checkpoint inhibitor. Now the question is, do we conjugate the immune checkpoint inhibitor onto the nanoparticle? We have done some studies testing that to see what the local effects or local benefits of the immune checkpoint inhibitor would be. But at the current form, we are administering the immune checkpoint inhibitor at its prescribed dose systemically. So what do these two pieces bring to the table? The Prussian blue nanoparticles generates tumor cell death, so it decreases tumor burden. It also causes tumor-specific antigen release and endogenous adjuvant release. Now, these effects are complemented by the immune checkpoint inhibitor that reverses immune cell exhaustion. Another piece or another modality that can be used is the addition of an immune modulator or an immune signal onto the nanoparticle. And what this does is it provides you an additional adjuvant if needed into the tumor microenvironment. So how would this work? You'd have the nanoparticle coated with the immune signal, and you'd administer photothermal therapy, and the tumors would die. So the presence of the exogenous immune signals in the context of dying tumor cells, and these immune signals that we work with are, for example, are toll-like receptor agonists. So this form of death in the presence of exogenous and adjuvant is fundamentally different than if you did not have this adjuvant, which would not create this additional boost to the immune system that we are looking to drive. So this is also very distinct from directly injecting the adjuvant into the tumor microenvironment and or systemic injection of the adjuvant. So if you put this immunological adjuvant systemically, there would be a biodistribution of it, and there might be a blanket activation of the immune system, but we really want to focus the effects of the boost to the immune system in the tumor microenvironment, which is why the nanoparticles being coated with the immune signal provides this added benefit. What else does the nanoparticle confer? It allows you to capture the released antigens in the tumor microenvironment. So the dying tumor cells release the antigens that bind to the nanoparticles. So how would this look? So take, for example, the dying tumor cells releasing these red antigens, just in a cartoon depiction. The nanoparticles provide a surface to absorb the antigens released from the dying tumor cell. Now you can imagine that if you didn't have the nanoparticle, you would just have the dying tumor cells and the released antigens and endogenous adjuvants. And that form of death is fundamentally distinct from this antigen capture modality of the Prussian blue nanoparticles or any nanoparticle that we use. In fact, the importance of antigen capture was first described in a paper around 4 years ago, where people use radiation therapy in combination with immune checkpoint inhibitors for melanoma. What they found was administering an antigen capturing nanoparticle improved the abscopal effect of this combination therapy. So even there, where they were using radiation therapy, they found that addition of this exogenous agent that serves as a platform to which all these dying tumor cells release its contents and then binds to was advantageous to generating an abscopal effect. So now that we've described what are the phenomena that the nanoparticles and the nanoimmunotherapies contribute to, there are some advantages and some limitations that I'd like to describe. So similar to other cytotoxic approaches such as radiation therapy, ablative therapies, chemotherapy, PTT releases tumor antigens as well as endogenous adjuvants. But the advantages are the ability to deliver an anchored immunological signal in the tumor microenvironment and to serve as a surface to capture the antigens released in the tumor microenvironment. However, given that you have these advantages, there are some limitations of using light-based therapies. For one, there is a depth of penetration issue. So when we talk about light at a therapeutic dose to activate the nanoparticles, they don't penetrate to infinity. It actually penetrates a maximum up to a centimeter, depending on what wavelength of light is used. So there is a depth of penetration issue. So in order to overcome this, you can use a fiber optic laser through a catheter that then allows the light source to be present right in the tumor microenvironment. And this can be done in a minimally invasive manner. And there is a whole modality known as laser interstitial thermal therapy, where we could use that existing infrastructure to then inject the nanoparticles in the tumor microenvironment and then leverage the advantages of nanoparticle-based PTT. So what are our envisioned clinical uses? At this point, we don't think that we would get approval to start our nanoimmunotherapy as a first-line treatment. So we actually would look to get into clinical trials as an experimental therapy for patients that are radiation dose limited. So where they cannot go through any further rounds of any radiation or chemotherapy, for example, or if tumors are inoperable. So if you are surgically unresectable tumors, you could consider using them as candidates for photothermal therapy. You could also use the interventions pre-surgically so that you can then jumpstart the immune response that can then, if there's any residual tumor left over, that can be cleared with a surgical intervention after this pre-surgical nanoimmunotherapy. So now for the context of this particular, the concept for expanding upon the concepts I presented, I'm going to be talking about our work in the realm of neuroblastoma. We've done work in other cancer types. I'm showing you neuroblastoma because it's the most developed workflow that we have had in our research group. And it represents a continuous story that we've been building over the past 7 years. So for those who are uninitiated, neuroblastoma is the most common pediatric solid tumor outside the central nervous system. So the moment you use the word neuro, most people think that neuroblastoma presents in the central nervous system, but it is actually the most common pediatric solid tumor outside of that location. And they present in the abdominal cavity and over the adrenal gland. So it's actually deep in the abdominal cavity towards the back or retroperitoneal. Now, why are we interested in neuroblastoma? It is because besides the prevalence, high-risk neuroblastoma, the survival rates have not really budged over the last few decades. It used to be 30 to 40%, but currently the overall survival is between 40 and 50%, despite intensive multimodal treatment options, which involves surgery, radiation, chemotherapy, stem cell rescue, other monoclonal antibody immunotherapies. Despite that, this is the range in which overall survival has landed. So that's why we believe that there is a good case for using the nanoimmunotherapy for treating neuroblastoma. So I'm going to develop the idea of how we've made the nanoimmunotherapies for neuroblastoma with these questions. The first is, what is the effect of PBNP PTT on tumor growth? What are the effects of this PTT plus immune checkpoint inhibitor on tumor growth? How do the tumor cells die after PBNP PTT? This was expanded upon extensively in the second lecture, so I'll only briefly remind all of you what we did. And then what is the effect of adding an immune signal onto the nanoparticle during PBNP PTT? And then finally, how does the ensemble, the nanoparticle, the immune adjuvant, and the immune checkpoint inhibitor work in tandem to potentiate a robust abscopal effect? So what's the effect of PBNP PTT on tumor growth? So we worked again with this neuroblastoma, just to refresh you, a most common solid tumor of childhood outside the central nervous system. We used a syngeneic mouse model of neuroblastoma. So neuroblastoma tumor cells transplanted into fully immunocompetent animals. We first tested the feasibility in subcutaneous models. So we injected the tumors subcutaneously, and we allowed them to be palpable, which is around a volume of 5 millimeters in one dimension. And then you can see out here, this was the tumor. we administered Prussian blue nanoparticles, a single intratumoral injection at 2.5 milligrams per kilogram. That corresponds to around 50 micrograms of the nanoparticles in the tumor microenvironment. And then we irradiated these tumors using an 808 nanometer laser at various laser fluences for 10 minutes. So these were the ranges we tested at that time. But more recently, we have settled on a fluence rate closer to 1.5 to 2 watts per centimeter square. Now, in terms of the results, if you have untreated neuroblastoma subcutaneous tumors, they progress over time. The use of just the laser without the nanoparticles have no statistically significant difference in tumor progression. But what we were able to see is that this combination of the laser plus the Prussian blue nanoparticles was able to bend down the tumor progression curve. So what we could see from these initial testings from about 7 years ago is that PBNP-PTT decreases tumor growth rates, but there was no long-term survival. So this is not totally uncommon or unexpected, because the primary driver of the therapeutic response at the time was just the cytotoxicity or the cell killing of the Prussian blue nanoparticle-based PTT. So then we asked the question, with all these immunotherapies being developed in the literature, could we combine them in a nanoimmunotherapy and see the effects on tumor growth in this exact same model? So once again, we use the same subcutaneous model of neuroblastoma. This, again, to refresh people, was we subcutaneously injected immunocompetent neuro 2A cells in an immunocompetent mouse and started the treatment when the tumors were palpable. So here, on day 0, we administered the PTT at that same thermal dose conditions of 2.5 mg per kg intratumorally with an 808 nanometer laser for 10 minutes on day 0. And over here, we use a laser power density of between 1.5 to 2 watts per centimeter square. And then we systemically injected immune checkpoint inhibitors at a dose of 7.5 milligrams per kilograms. This was anti-CTLA-4, clone 99, and we gave 3 administrations of it on days 1, 4, and 7. So the day after PTT, and 2 further administrations. Now, I must point out here that if you really wanted to optimize the anti-CTLA-4 dosing regimen, people have used multiple weeks of this dosing regimen to see the benefits of it. But at this point, we wanted to see what that front-loaded benefit of this nanoparticle-based PTT with the immune checkpoint inhibitor was. So as you can see here, in these subcutaneous tumor-bearing animals, we can see the bioluminescence going away and decreasing. And then in this combination therapy-treated mice, by day 12, that there's no bioluminescence, and even the scar that happens because of the thermal injury resolves. Now, in terms of survival, how did that translate? If you look at the untreated neuro 2A tumors, they succumbed very quickly post-palpable tumor to tumor burden. Now, we added anti-CTLA-4 at that dosing regimen that I described in the previous slide, and we see that we have about 1 in 8, or around 12.5% long-term survival. Now, this is very common with what we would see in the literature in terms of some of the response rates for specific tumor types. When we added PTT, as I'd shown you in that very first question answer, that there is an increase in median survival, where this median survival is higher than this, but we were not able to see any long-term benefit of administering PTT. But the combination of the two, the PTT plus the anti-CTLA-4, generated significantly higher long-term survival in this combination therapy-treated group. Now, I am just highlighting the first 25 days post-treatment to see the individual trajectories for the different treatment modalities. But we were able to project out up to day 100, where the animals continue to survive tumor-free and without any progression of observable tumor, as measured by bioluminescence imaging and physical inspection. So we could conclude from this study, which has been published in this paper, that PBNP-PTT plus this anti-CTLA-4 results in long-term survival in a significantly higher proportion of animals relative to all the other individual treatments and controls. So that was very encouraging for us. Then we asked the question, what immune cell players may be important in driving the responses? So for this, we did depletions of specific subsets in the animals. So how is this done? You administer these depleting antibodies that deplete either CD4, CD8, or other targets in the animals. And you verify that by taking blood draws from the animals and then testing a decrease in the actual populations compared to undepleted blood. And we were able to see that if we depleted the CD4 or the CD8 population, the survival rate or the protection that we would see from the combination therapy was abrogated, and the animals rapidly succumbed to tumor burden in the absence of CD4 or CD8 cell populations. So we could see that the depletion of these cell populations abrogated the observed therapeutic responses. Next, we tried to see if there was any aspect of protection against tumor rechallenge. And so in this study, we took the long-term surviving animals and basically subjected them to a rechallenge 100 days after the initial palpable tumor was treated. And as control, we used age-matched naive mice that were treated with the tumor for the first time, so they did not see the tumor at any point in time. And as to be expected, these particular naive animals showed tumor progression as measured by the black trajectory here. And as you can see, there's a slight decrease. It may look like a straight line, but there's a decrease in the tumor burden as measured by this bioluminescence in the rechallenged animals. And so 100% in this particular modality of animals survive long-term for a further 60 to 90 days after tumor rechallenge compared to the naive animals that rapidly succumb to tumor challenge. So this was very encouraging for us because it showed or demonstrated to us that the long-term surviving mice treated with our nanoimmunotherapy exhibited protection against tumor rechallenge. Next, we asked the question. So we had worked on what are the cytotoxic effects of PTT. How can they be combined with this anti-CTLA-4 immune checkpoint inhibitor? Now, to answer your question, why did we choose anti-CTLA-4? It is because at that time, that was the most developed immune checkpoint inhibitor when we were doing our studies. We have subsequently looked at other immune checkpoint inhibitors such as anti-PD-1, anti-PD-L1, and we do see a response benefit with anti-PD-L1, but not so much with anti-PD-1. Now, those were in terms of the pieces. Then we asked the questions, how do the tumor cells die after PBNP-PTT? And so I will not go over this in great detail because you can watch the recording of my second talk, but based on the thermal dose administered in the tumor microenvironment, if the thermal dose was too low or too high, you have immunologically silent tumor cell death. And at the optimal tumor thermal dose, we were able to generate immunogenic cell death. And this provided long-term protection in these, when administered at this tumor dose in these treated animals. So the next question was, okay, so we can now generate PBNP-PTT at the optimal thermal dose. It works well with anti-CTLA-4 immune checkpoint inhibitor. Can we boost the response rates even further? Because remember, we're still working with a single tumor model and subcutaneous. We have to move towards being able to have a feasible shot at treating high-risk neuroblastoma, which presents with disseminated disease at initial diagnosis. So we then asked the question, what is the effect of adding an immune signal during PBNP-PTT? So in this study, we again used the subcutaneous model of neuroblastoma and commenced our treatments when we had 5 millimeters tumors in one dimension. For this study, we coated the Prussian blue nanoparticles with a layer of CpG oligodeoxynucleotide. So this is a TLR9 agonist, and we coat them on using layer-by-layer technology. Now the question is, why do we use CpG and why not any other TLR agonist? Well, there was a very seminal paper published by Carpentier et al. in 2000 or I believe 1999, where they were able to inject CpG in Neuro2A tumor-bearing animals and see a survival benefit. But once again, in that context, the CpG was administered systemically or intratumorally. But we wanted to see if we can anchor the CpG onto the nanoparticles, and to achieve that, we use what is known as layer-by-layer technology. So in a very simple manner, these blue cores of Prussian blue are negatively charged. And then we coat them with a layer of linear polyethylene imine, which is a positively charged polymer that is used for complexing, and it's also used in transfection or delivery of nucleic acid agents to targeted cells. So we use linear PEI as opposed to the branched PEI that is available, because linear PEI has a lower toxicity than branched PEI and a much easier synthesis profile. But as a result of decreasing toxicity and molecular weight, you also have decreased binding capacity of the nucleic acid. So that's a trade-off that you have to make when synthesizing your nanoparticles. So what was the treatment regimen that we used? So we used CpG PBNPs for PTT, and we administered it, again, 50 micrograms in the tumor microenvironment, or 2.5 mg per kg intratumorally, with the 808 nanometer laser at 1.875 watts per centimeter square for 10 minutes. Now, that was done on day 0, but in some studies where we were testing the effects using CpG PBNP, we boosted the animals with 2 subsequent boosts of just CpG PBNP on day 2 or day 3 and day 5. So when used, we either use PBNP PTT or CpG PBNP PTT on day 0. And when CpG was used, we used the CpG PBNP without the ablative piece on 2 specific doses based on the dosing regimen 1 week after the initial treatment. So what is the benefit of adding CpG PBNPs compared to just the free PBNP PTT? As you can see here, untreated animals or vehicle-treated animals succumb rapidly to tumor burden after it had palpable tumors. Now, if you administer a free CpG into the tumor microenvironment, there is a median survival benefit here, but you don't see any long-term survivors. If we put CpG onto the Prussian blue nanoparticles and delivered them 3 times without PTT, you do have an, again, improvement over the free CpG. So that's the benefit of using a nanoparticle for immune signal delivery out here. And we saw one animal surviving longer, but it eventually succumbed to tumor burden around day 70. And then we use PBNP PTT, we have the significantly higher improvement in median survival compared to just the CpG PBNP, free CpG, and untreated. As you can see here, compared to some of the previous studies that I presented with the same model, you can see that the median survival is way higher than what we have observed in the past. And that was just because we are becoming more and more informed as to the thermal dose of what was the CpG, PBNP PTT being administered. So in the initial studies, we would just administer PTT without paying careful attention to the thermal dose. And once we had done those optimization studies, we administered this PBNP PTT at that specific immunogenic thermal dose and were able to boost median survival. Now, how do these pieces work in tandem? When we use CpG PBNP PTT on day 0, we were able to see, plus followed by 2 further boosts of CpG PBNPs on day 2 and day 5, we were able to see that 70% of the animals showed long-term survival and complete tumor regression. So now keep this in mind that when we first used PBNP PTT plus anti-CTLA-4, we only saw about 60% long-term survival. But adding this immune adjuvant and optimizing the thermal dose, we were getting up to 70% long-term survival. So CpG PBNP PTT results in long-term survival in a significantly higher proportion of animals compared to these controls. So this is very encouraging to us, saying that the importance of adding an immune signal. Now, once again, we took these long-term surviving mice from the previous study, and we re-challenged them with the tumor. And as we saw with the combination of PTT plus anti-CTLA-4, 100% of long-term surviving mice were able to reject tumor re-challenge compared to naive mice that succumbed more rapidly to tumor burden. So there was protection against tumor re-challenge in these CpG, PBNP, PTT treated animals. So we wanted to build on this a little further. And so we saw results in the Neuro2A model of neuroblastoma. And we all understand that there are inherent limitations of working with a single tumor model. So we wanted to verify if the CpG, PBNP, PTT would work in a Th-Mekin model of neuroblastoma. So in this particular study, we worked with single and two tumor models. So we had either one tumor on the mouse flank or two tumors on opposing flanks, where we administered CpG, PBNP, PTT to one tumor and not the second. So this is a paper coming out relatively soon. It's in press. And here, I'm sorry I got the name wrong, but the journal is correct, and all the DOI of this particular publication is all correct, except the first author. And so we see over here with CpG, PBNP, PTT in this more aggressive Th-Mekin model of neuroblastoma, what ends up happening was this generates tumor cell priming. So the tumor cells themselves upregulate MHC1, MHC2, and co-stimulatory molecules. And as I presented before, we administered them at a thermal dose, this PTT, to generate immunogenic cell death. Now, there's a whole panel where we study the T cell activation and robust memory generation. So we treated the tumors, they survived long-term, we re-challenged the tumors, and they rejected it. And then we also saw an abscopal effect in a tumor model. And more importantly, in the animals that rejected the tumor re-challenge, we took them and isolated the T cells from the spleens, not the tumor-draining lymph nodes. So presumably, it would be even less in population or numbers in the spleen compared to a draining lymph node. And we co-cultured these T cells with tumor cells, and we were able to see a tumor-specific response by these T cells. So now this is expanded upon in this particular publication, and I would encourage everyone to read it. So now we have shown how these pieces work together. PTT causes cytotoxicity. It can generate immunogenic cell death. Addition of an immune signal provides a survival benefit as well as memory, as well as combination with anti-CTLA-4. So we wanted to challenge the model a little more to see what the benefits would be if we went into animals bearing multiple tumors. So we asked the question, does this local PBNP-PTT combined with systemic immune checkpoint inhibitors potentiate a robust abscopal effect? Because remember, our target population is still high-risk neuroblastoma, where you have tumor dissemination. So for this particular study, we didn't use a single subcutaneous model of neuroblastoma, but a synchronous model of neuroblastoma. So what do I mean by that? So you would have tumors, an animal that was subcutaneously implanted with two palpable tumors. And then what we did was we injected either the PBNPs or CpG PBNPs onto the single tumor and not the second one. We irradiated the first tumor that was injected with the PBNPs or CpG PBNPs with the laser to deliver PTT. Now, subsequently, in the peritumoral environment, we systemically, we administered anti-CTLA-4 and or the CpG PBNPs. And we monitored tumor growth and survival. So you have PBNP, CpG PBNP, PTT, and systemic anti-CTLA-4. So there is this ensemble, group of cytotoxic and immunotherapies put together in this nanoimmunotherapy. So let's go over what the results were. If you look out here, so these are animals bearing two tumors. So the vehicle group rapidly succumbed to tumor burden. So the two tumors were untreated, they progressed. Anti-CTLA-4 roughly matched at the dosing regimen that we used, roughly matched untreated animals. When we used PTT, what happened was the primary tumor went away, and that provided some decrease in tumor burden, and while the secondary tumor progressed. And so we would have an increase in median survival. When we added CPG onto the Prussian blue nanoparticles and treated them onto one tumor with those CPG PBNP boosts, we were able to see an even increased median survival compared to PTT by itself, even in this two-tumor model. When we use PTT without the CPG and the anti-CTLA-4, what we were able to see is that most primary tumors, if not all, completely were eradicated. And in a fraction of them, around 1 in 5 of them, we would actually see regression in the secondary tumor. And so that was very encouraging because it showed to us that there was a robust abscopal effect. And now when we put all the pieces together, the ablative piece, the immune signal, and the systemic immune checkpoint inhibitor in this two-tumor model, we were able to see that all the pieces worked in concert to give nearly 60% long-term survival. So it was interesting to see that the primary tumor would go away and the secondary tumor would look like it was progressing in some of them. And in some cases, they progressed. But in other cases, they basically started to increase in size, and then we would see a decrease, a rapid decrease in tumor burden. So that was very encouraging for us, suggesting that that initial infiltration or increase in tumor size was a consequence of the tumor, the immune system infiltrating the secondary tumor and mounting a response. So we can conclude from this published study that CPG, PBNP, PTT plus anti-CTLA-4 immune checkpoint inhibitors resulted in the highest long-term survival relative to control. Excuse me. So in terms of looking at whether the animals had any protection against tumor rechallenge, similar to the other studies that we showed before, when we took the long-term surviving animals, the animals were protected against tumor rechallenge in 100% of the long-term surviving mice, suggesting some component of immunological memory. More importantly, we again depleted CD4 and CD8 subsets, and we verified depletion through flow cytometry of the blood. And we observed that the depletion of CD4 and CD8 abrogated the protective responses of the nanoimmunotherapy, and these animals progressed similar to untreated animals. So this was very encouraging for us because it shows to you the development of the body of work wherein we started with an ablative therapy and started adding immune pieces to it, as well as existing immunotherapies to boost treatment responses for a disease that currently has a very poor prognosis, which we are building towards. So just to summarize in this particular instance, what the effects of the PBNP PTT-based nanoimmunotherapies are doing in terms of the questions we asked. So what is the effect of PBNP PTT on tumor growth? We see that we observed that it was able to decrease tumor growth because of the cytotoxic effect of the PTT. Then we asked the question, what are the effects of the PTT plus immune checkpoint inhibitor on tumor growth? We were able to see complete tumor regression, long-term survival, and a protection against tumor rechallenge in the majority of the animals. Then when we asked the question how the tumor cells die after PBNP PTT, we were able to conclude that they can undergo immunogenic cell death depending on the thermal dose administered to the tumor cells. We asked the question, what is the effect of adding an immune signal without the immune checkpoint inhibitor during PBNP PTT? And in the single tumor model, this CPG PBNP PTT could elicit long-term survival and protection against tumor rechallenge. And then finally, we asked the question, does this PBNP PTT or CPG PBNP PTT plus systemic checkpoint inhibitors potentiate a robust abscopal effect? And the answer is yes, we've observed this in the tested neuroblastoma models. I'm encouraged to let you know that in other studies that are under review and that have been currently being performed in the lab, that we see these effects of the combination therapy even in these other animal models as well. So to just give you a summary, PBNP light-based nanoimmunotherapy, I presented them, how we can use them to modify the tumor microenvironment and potentiate a robust anti-tumor immune response. So what are the pieces? They are basically the Prussian blue nanoparticles for PTT that co-deliver immune signals in the tumor microenvironment, combined with systemic immune checkpoint inhibitors. So what are the effects that work in concert to generate a protective response? Through the nanoparticles, we generate tumor cytotoxicity, tumor antigen release, endogenous adjuvant release, and exogenous immune signal delivery in the tumor microenvironment. And these effects are complemented by the effects of the systemic immunotherapy, in this case, immune checkpoint inhibitor. So this is promising, and where are we going with this particular platform? We are now applying the nanoimmunotherapy in orthotopic neuroblastoma models. Now recall, neuroblastoma is not a subcutaneous tumor. It is a deeper tumor, and it presents retroperitoneally. So we are trying to study the effects of our nanoimmunotherapy in these deeper tumor models of neuroblastoma. This will take some time because it requires some surgical procedures to be done to implant the tumors. And another key piece of doing that is that if the tumor is deeper, you can't use an external beam laser. So we are delivering the light interstitially. So this required us to reverse engineer the technology a bit, because you have the technology existing in the clinic with LIT, but they are not small enough to work with animals. So we are actually working with catheters that are tiny, the smallest size bore, so a higher gauge that will allow us to snake a catheter that is around 0.5 millimeters or 500 microns wide. So that is allowing us to deliver light to 5 millimeter tumors. And so that's with trying to translate the technology further. We're also using other tumor models where I've mentioned to you, we've worked with melanoma, and some of the work that I did with breast cancer will be presented here in Brazil at the Brazilian Breast Cancer Symposium tomorrow. And with that, I'd like to acknowledge the immense work done by the team. Here is my current team of superstars. And then I'd like to acknowledge the hard work done by the team alumni, in particular, Juliana Cano Mejia and Preeti Balakrishnan, who started this work. We've had immense collaborators that provide us the clinical context at the Children's National Hospital, Dr. Catherine Bollard, Dr. Russell Cruz, and many of the others that are too many to list here. And finally, I'd like to acknowledge George Washington University for giving us the resources and the funding sources for supporting the work we do in our lab. And with that, I'd like to again thank all of you very much for your attention. I look forward to having some questions in the question answer session. And please feel free to get in touch with me if you need any more information on the work we are doing going forward. Thank you.
Thanks, Rohan, for the excellent talk. We have some questions here by Dr. Rony Mioto from Federal University of ABC. Rohan, this kind of chromophorus has already been used successfully in photodynamic therapy. Can be part of these results be attributed to cell death induced by PDT? Instead, have you checked, for example, oxygen singlet production to access this issue? PDT is also known to stimulate the immune system.
So the short answer is this work is in progress right now. I'm working with a collaborator from Roswell Park Cancer Institute where PDT was invented. And so we are trying to understand whether there are other PDT effects of our photothermal therapy as well. So the short answer is we haven't really, this is work in progress, and I don't have anything to disclose on that front presently, but we are actively working on it, and it's an important point. But I also want to distinguish, the chromophore versus the nanoparticle identity. So when we say chromophore, and it may be as a manner of speaking, but the general opinion is like when we talk chromophore, we're thinking small molecule. I just want to, stress the importance of the fact that this is a nanoparticle that has a colloidal identity, and the benefits of that colloidal entity is what we're trying to also explore. Now, admittedly, we don't know the reactive oxygen aspects, species-based aspects of this therapy just yet. But this is actually a very fantastic question and timely because we are working on it.
Thanks, Rohan. Rohan, I'm curious about the magnetic properties of these nanoparticles because you have iron 2 plus and iron 3 plus. Have you checked the magnetic properties of this? It is an anti-ferromagnetic, ferrimagnetic. What is this? What are the properties, magnetic properties of this material?
So it does. So when you cool it down, then it starts to behave magnetically. At room temperature, it does not have any magnetic properties. We have incorporated in the interstitial space, elements such as gadolinium and manganese, and we've used it for molecular imaging. But then again, there is no magnetic property per se, except for MRI. But we've done supercooling of these materials, and you should look up the paper by Dumont et al. From my group in 2014, I believe, or 2013, December, which came out, which talks about the squid magnetometry using these particular Prussian blue nanoparticles by themselves and with the addition of gadolinium.
Okay. And when you were talking about the PTT not showing a robust effect, the animals being dying, right? You did not mention exactly what was the thermal dose. So you could control the thermal dose, and depending on the thermal dose, you can have a better result with PTT, right?
Yes. So in our initial studies, we didn't control thermal dose. We used the setting that the more is better. So that's why I gave you a range of 1.5 to 4 watts per centimeter square. Yeah. But then subsequently, when we did a deep dive, we understood that we want to be, and this really depends on the tumor type, and that's why it's helpful to optimize it. So it would be an opportunity to maybe, if we had enough of a biopsy surgically, you could actually do a single cell suspension and then do these optimization studies in vitro or ex vivo before then determining if the candidate needs to go back for a second round where we're going to administer PTT, or we'd be able to find the thermal dose for that particular tumor. But no, at the point when we started, it was more like a blanket and a different perspective rather than a nuanced perspective that we have now on thermal dose.
Okay. And can you make a comment about the size of the tumors that you are choosing to start to do PTT? Because you told that it's around 5 millimeters. Have you tried, you're using 5 millimeters in the sense that the laser will pass like homogeneous inside the tumor? Is that the idea? So you cannot work with larger. Can you make a comment on what is the size of the tumor that you usually can work with for this type of therapy, for the design of the experiment?
So you bring up a very important point of feasibility, because clinically you can just use the LIT system and you can just slowly withdraw the catheter and irradiate the whole tumor volume. You can place the catheter and then irradiate it. But for us, from an experimental standpoint, it's hard to do that. So we had to pick a size that matched with our laser dimensions. But to answer your question, that's just a practicality of administration. When you go higher in tumor burden, like say you start with 1 centimeter tumor. So we've seen results with as high as 7 to 8 millimeter tumors. So that would require a little bit of a regional effect of the anti-tumor immune response. Those work actually quite well for local control. But when you go to 10, then you have an existing amount that is unirradiated, the tumor, and that already has an immunosuppressive environment. That coupled with other infiltration that comes in may actually doom your therapy much more. So I think you have to try and irradiate and get as much cytotoxicity as possible. If some areas are left out, the local immunity is good enough to do local tumor control. But if it's too big, it's already representative of a higher suppressive population of the tumor microenvironment. And then that just goes out of control from that point. So
do you have do you know any paper that had discussed this a little bit more? Because the size would matter here. So would there be a specific volume that you can immunologically activate that would be enough for this to work? I have never seen in the literature something regarding PTT. Is this a question that is relevant for thinking about?
Absolutely. And I think this is important for us to have this discussion. The person that I was talking about from Roswell Park, Dr. Gal Schifferstein, he's working on like light distribution in larger tumor volumes. But I think the anti-tumor immune effects of PTT have only been uncovered in the past 3-4 years. And if you look a lot at the literature, the majority of work is we make a new material and show proof of concept. So it's more like breadth with novel materials. You are really talking about depth. And so I think it's very important for the audience to realize that these simple questions can be answered. The way we try to answer it is by injecting multiple tumors. So we have actually done 3 tumors, and 1 tumor can take care of the other 2 as well. But having all of them in one consolidated standpoint, that has not been done. And it may not even be realistic, because if you were to project what size of tumor we would talk about a human, it may not even be something feasible or representative as well. But you can inject multiple nodules and study the same thing similarly. So if you have 200 millimeter cube volume versus 300 millimeter cube, you can do 3 tumors of 100 millimeters and so on. So, but not one single tumor that is that huge.
So in the children, usually the tumor size is around which one? Do you know?
So actually, in neuroblastoma, the tumor sizes are larger. So we're talking about a few centimeters. So they are about the size of a tennis ball or a golf ball. Like that level of resection happens. But that is completely, on the cards for laser interstitial thermal therapy. I've talked to neurosurgeons here, and they routinely ablate that size of lesions in the body. So I'm not sure about a tennis ball, but for a golf ball for sure is something we could look at. So we would have
frequency ablation, something like that.
Yeah, right. And so we would have to look at a combination of surgery with PTT in that setting, maybe in the same treatment. Like, if you do surgery and then this ablative aspect as well to boost the immune response.
Rohan, Mioto has another question, Dr. Mioto. There must be, regarding the concern with PDT and PTT, he mentioned the following. There must be, since you are using Prussian blue, and its absorption wavelength is 798 nanometers, exactly what you're using. My suggestion is to try to measure singlet oxygen generation to answer this question. That's his comment here.
Well taken, and yes, we will look at that as well, the singlet oxygen.
Well, I guess we are ending our school. I really appreciate your talk, Rohan. Thanks again for being with us in the second advanced school of nanomedicine. For those that would like to see another presentation by Rohan, tomorrow he would be in the workshop on nanomedicine and immunotherapy from the Brazilian Breast Cancer Symposium. I put in the chat, I put information about how to register there. And basically it was really great to have you here, Rohan. I hope you have enjoyed too.
Phenomenal experience. Thank you so much. And all the Brazilian colleagues, and muito obrigado. I really appreciate this opportunity, and be well, be safe, and come visit Washington, D.C. And I hope that I can meet and we can talk these matters about singlet oxygen measurement over coffee and more in the near future. So thank you so much, everybody.
Thank you. And so I thank all the speakers, Rohan, Robert, and Carlos, for the excellent talks and the mini courses. And of course, I thank you all, the participants that have been watching and discussing with those excellent scientists about cancer nanomedicine. Thank you all. And as Rohan said, stay safe. Bye-bye, Rohan. Bye, guys. So we are ending the second.